Dr. Mishra ji is the assistant professor and scientist in the Department of Dravya Vigyan, Patanjali Ayurved College, Haridwar. He is a gold medalist in MD and has more than 80 national and international publications in medicinal plants. Let us all give him a warm welcome. Dr. Rajesh Mishra ji will be giving his talk on nature's signature in Ayurveda with an ethnopharmacological approach.
ओम ओम परम श्रद्धेय प्रातः स्मरणीय पूज्य स्वामी जी महाराज, परम पूज्य आचार्य जी महाराज की सूक्ष्म उपस्थिति को प्रणाम करते हुए और मेरे यहाँ पे उपस्थित समस्त शिक्षकगण और रिस्पेक्टेड गेस्ट उनको प्रणाम करते हुए मैं आज का जो मेरा टॉपिक है नेचर सिग्नेचर इन आयुर्वेदा विद एन इथनोफार्माकोलॉजिकल अप्रोच, आई वुड लाइक टू प्रेजेंट माय टॉपिक इन अ 3 वे because ethnopharmacology is a very important and a unique topic and also subject. In ancient Ayurveda, a lot of evidences are present for the uses of the ethnopharmacology. As we know, what is the means of Ayurveda? Ayurveda is made from 2 words. The first one is Ayu and second is Ved. So the means of Ayurveda is the science which is dealing from the life. A science of Ayurveda, a life of Ayurveda. It's a science of life, and the main principle of the Ayurveda is to protect from the disorder of the and to heal the disorders. अगर हम इसको सिंपल में बात करें, एक बहुत ही अच्छा कोटेशन है आयुषो वेदा आयुर्वेदा, अर्थात जो आयु का वेद है वो आयुर्वेद है। और आयुर्वेद में बहुत सारी चीजें ऐसी डिस्क्राइब की गई हैं जो कि पर्टिकुलरली हमारे नेचर से कहीं ना कहीं एक साइन के रूप में या सिग्नेचर के रूप में बिलोंग करती हैं या पर्टिकुलर इस चीज को प्रेजेंट करती हैं। सेम थिंग इज ऑल्सो डिस्क्राइब इन पुराणाज, और वो एक सेंटेंस बहुत ही अच्छा पूज्य स्वामी जी महाराज बोलते हैं, यत पिंडे तत् ब्रह्मांडे, अर्थात जो कुछ पिंड में है वही ब्रह्मांड में है, जो कुछ हमारे बॉडी के इनसाइड है सेम सिचुएशन सेम कंडीशन हमारे बॉडी के आउटसाइड है, यह इस पूरी यूनिवर्स में है। ऐसा ही एक जो प्रिंसिपल है जो आयुर्वेदा में डिस्क्राइब किया गया है, उस प्रिंसिपल का नाम है पंच महाभूत सिद्धांत। यहाँ पे बहुत सारे आयुर्वेद के स्टूडेंट बैठे हुए हैं और वह इस पंच महाभूत सिद्धांत को समझ सकते हैं। इट मीन्स ये है कि our body and whole universe is made from the 5 basic elements like आकाश, वायु, अग्नि, जल एंड पृथ्वी। और इसी सिद्धांत के तहत एक ही बहुत ही यूनिक जो प्रिंसिपल है जिसको हम लोग DOS या डॉक्ट्राइन ऑफ सिग्नेचर बोलते हैं, जो कि 1741 में दिया गया था, 1491 में उसके 1491 में सबसे पहले दिया गया था, 1741 में उसको रिवाइज किया गया। थियोफ्रास्टस नाम के एक साइंटिस्ट हुआ करते थे, उन्होंने इस चीज को प्रपोज किया कि हाँ एक ऐसा प्रिंसिपल है कि जो कि ये दिखाता है कि जो कुछ हमारी बॉडी में है या जैसा कुछ हमारे बॉडी में ऑलरेडी है वैसी कंडीशन हमारे यूनिवर्स में भी मिलती है। और इसी आइडिया को लेकर के जो आज का ये टॉपिक है वो मैंने रखा हुआ है इस पे। अगर हम इसके हिस्ट्री की बात करते हैं तो जो सबसे हमारे पुरानी चीजें हैं, अगर हम आयुर्वेदा के पॉइंट ऑफ व्यू से बोलते हैं, दैट इज बृहत्रयी। बृहत्रयी मीन्स अ कलेक्शन ऑफ द 3 एंशिएंट ट्रीटेज ऑफ द आयुर्वेदा, आउट ऑफ देम चरक संहिता, सुश्रुत संहिता एंड अष्टांग। और उसके बाद में फिर सेम इसी तरह का जो पर्टिकुलर क्लासिफिकेशन है वो निघंटू में, स्पेशली इन राज निघंटू जो कि 14th सेंचुरी का एक ट्रीटेज है, और धन्वंतरी निघंटू जिसको कि हम 13th सेंचुरी का ट्रीटेज मानते हैं, उसमें डिस्क्राइब किया गया है। और सेम ऐसी ही कंडीशन 1491 से 1541 में जो थियोफ्रास्टस नाम के एक साइंटिस्ट थे उन्होंने डिस्क्राइब की। और उसके बाद में फिर 1575 से 1624 पे एक डॉक्ट्राइन ऑफ सिग्नेचर को प्रपोज किया गया था, जो कि एक रिलेशनशिप गॉड और मैन के बीच में एक रिलेशनशिप को क्रिएट करता है, उस चीज के बारे में बताया गया था। और देन इसके बाद में 1626 टू 1662 में विलियम कॉलिस नाम के एक साइंटिस्ट उन्होंने एक बुक लिखी, द आर्ट ऑफ सिंपलिंग एंड एडामीन एडन अबाउट द वॉल दैट क्यूरिंग इफेक्ट्स इन दे हैव द परफेक्ट सिग्नेचर ऑफ द हेड, जो कि सिग्नेचर ऑफ डॉक्ट्राइन या डॉक्ट्राइन ऑफ सिग्नेचर को कहीं ना कहीं आयुर्वेद के पंचमहाभूत सिद्धांत से जोड़ता है। यह आयुर्वेद में एक बहुत ही अच्छा प्रिंसिपल डिस्क्राइब किया गया जिसको बोलते हैं लोक पुरुष साम्य, उस पर्टिकुलर सिद्धांत से कहीं ना कहीं पे ये चीज मैच कराता है या जोड़ता है। और एक बहुत ही इंपॉर्टेंट बात है कि जैसे कि हम लोग बहुत सारी चीजें ऐसी हैं जो हर्बल मेडिसिन है डिपेंड करती है उनके करेक्ट आइडेंटिफिकेशन के ऊपर। तो सबसे पहले एक अगर हम बात करते हैं बॉटनी की, बॉटनी एक ऐसी विधा है जिसमें पर्टिकुलर प्लांट को नॉमेनक्लेट करते हैं, उनको क्लासिफाइड करते हैं। तो ऐसा ही एक मॉडर्न नॉमेनक्लेचर जो कि कार्ल वॉल लिनियस ने दिया था, उन्होंने भी अपने नॉमेनक्लेचर में इथनोफार्माकोलॉजी को बहुत ही इंपॉर्टेंट और यूनिक प्लेस दिया हुआ है। एक बुक जो कि 15 से 17 सेंचुरी में लिखी गई थी जिसका नाम था हॉटस मेलेवेरिकस, जो कि जॉन वॉन रीडे ने लिखी हुई थी, उन्होंने उसमें 743 प्लांट्स को डिस्क्राइब किया गया था। और वो 743 प्लांट्स जो थे उसमें डिफरेंट क्लिनिकल कंडीशन डिस्क्राइब की गई थी, और वो क्लिनिकल कंडीशंस बहुत सारी ऐसी क्लिनिकल कंडीशन थी जिनको इथनोफार्माकोलॉजी के बेस्ड के ऊपर पर्टिकुलरली नॉमेनक्लेट किया गया था। यहाँ पे जैसे आप देख सकते हैं बहुत सारी डिसीज्ड कंडीशन, इन्फ्लेमेशन, आर्थराइटिस, जीआईटी से रिलेटेड डिसऑर्डर, इन पर्टिकुलर डिसऑर्डर्स को एम करके उन्होंने इस प्लांट को नॉमेनक्लेट किया था, जो कि डेफिनेटली कहीं ना कहीं इस बात को बताता है कि इथनोफार्माकोलॉजी कितना इंपॉर्टेंट सब्जेक्ट है। तो यहाँ पे मैं कुछ ऐसे एग्जांपल्स प्रेजेंट करना चाहता हूँ, लाइक हॉलेरिना एंटीडेसेंट्रिका, एवरीबॉडी नो इट इज द कुटच, बट एंटीडेसेंट्रिका ये जो पर्टिकुलर प्लांट है जो कि लिनियस ने इसका नाम एंटीडेसेंट्रिका रखा हुआ है, इट मींस द पर्टिकुलर प्लांट यूज्ड फॉर द ट्रीटमेंट ऑफ द डिसेंट्री, और इसके ऊपर 13 पेटेंट ग्रांट किए जा चुके हैं, जो कि इस बात को दिखाता है कि ये पर्टिकुलर प्लांट की मेडिसिनल सिग्निफिकेंस कितनी है। और सेम ऐसे ही प्लांट है एक स्वामिडा फेब्रिफ्यूजा, फेब्रिफ्यूजा के मीन्स ये है इन आयुर्वेदा अगर बोलते हैं तो ज्वर शामक, तो फेब्रिफ्यूजा ऐसा प्लांट जिसका प्रयोग एंशिएंट टाइम से ज्वर की चिकित्सा में किया जा रहा है, और सेम कंडीशन जो है लिनियस ने अडॉप्ट करके इस पर्टिकुलर प्लांट को स्वामिडा फेब्रिफ्यूजा नाम दिया हुआ है। ऐसे ही एंडोप्रोगॉन जोरांकुसा, इन संस्कृत जोरांकुसा मीन्स द पर्टिकुलर प्लांट विच इज यूज्ड फॉर द ट्रीटमेंट ऑफ द फीवर, एंड आइबोमिया परगा, परगा मीन्स लैक्जेटिव एक्टिविटी। तो ये ऐसे प्लांट है जिसका उपयोग या तो फीवर की चिकित्सा में या लैक्जेटिव एक पर्टिकुलर प्रॉपर्टी को लेकर के इन पर्टिकुलर प्लांट्स के नाम दिए गए हैं। ऐसा ही एक तीसरा प्लांट है जिससे आप सभी लोग जानते हैं पेपावर सोमनीफेरम, जिसको अफीम बोलते हैं। अफीम एंशिएंट टाइम में इसका बहुत ही यूज किया जाता था, और अभी भी निद्राजनक के रूप में अफीम का बहुत ही ज्यादा प्रयोग करते हैं। एंड सेम कंडीशन टाइलोफोरा अस्थमेटिका, टाइलोफोरा एंटी अस्थमेटिका और टाइलोफोरा अस्थमेटिका बोथ आर सिनोनिम्स। तो ये जो पर्टिकुलर प्लांट है ये एंटी अस्थमा या अस्थमा के ट्रीटमेंट में किया जा रहा है। फिर इस नेक्स्ट प्लांट है सेंट्राथीरम एंड हेलमिंथिकम, ऐसा प्लांट जिसका प्रयोग वर्म इन्फेस्टेशन के लिए या कृमि रोगों की चिकित्सा के लिए किया जाता है, और इसके ऊपर अभी तक बहुत सारे ऐसे 120 नोन कंपाउंड्स हैं लाइक फ्लेवोनाइड्स, फैटी एसिड, स्टेरॉल्स, जो पर्टिकुलर किसी ना कहीं इसकी मेडिसिनल वैल्यू को ट्रू करते हैं या इसकी मेडिसिनल वैल्यू को पोटेंट रूप से दिखाते हैं। और एक प्लांट है फाइलेंथस यूरिनेरिया, इस पर्टिकुलर प्लांट्स का प्रयोग जितने भी तरह के रीनल डिसऑर्डर्स है उन डिसऑर्डर्स के ट्रीटमेंट के लिए किया जा रहा है। और ये जो यूरिनेरिया शब्द है इसका मतलब यही है कि एक ऐसा प्लांट जिसमें डायूरेटिक प्रॉपर्टी होती है। तो ये जो बॉटनी में बाइनोमियल नोमेनक्लेचर दिया गया है लिनियस के द्वारा, उसमें इन्होंने ये तो सिर्फ कुछ एग्जांपल्स थे, ऐसे बहुत सारे प्लांट्स को उन्होंने या बहुत सारी ऐसी प्रॉपर्टीज को उठाया है जिसको आधार मान करके, जिसको बेस मान करके उन्होंने डिफरेंट टाइप के जो नोमेनक्लेचर हैं वो प्रोवाइड किए हैं। सेम ऐसे ही कंडीशन अगर हम एंशिएंट नोमेनक्लेचर की बात करते हैं, आयुर्वेदा की जो हमारा इंडियन ट्रेडिशनल सिस्टम ऑफ मेडिसिन है उसमें बहुत सारे ऐसे एग्जाम्पल हैं, उसमें से मैं कुछ आपके सामने यहाँ दिखाता हूँ। लाइक कैलोट्रोपिस जायंसिया, इस पर्टिकुलर प्लांट का नाम है इन संस्कृत इट इज नोन एज अर्का, बट इसका एक सिनोनिम है कुष्ठारी, कुष्ठ मीन्स लेप्रोसी, अरी मीन्स ऐसा पर्टिकुलर प्लांट जो लेप्रोसी को दूर करता है। तो ये इस पर्टिकुलर प्लांट का प्रयोग जितने भी टाइप के स्किन डिसऑर्डर है लाइक लेप्रोसी, ल्यूकोडर्मा, इन सब के ट्रीटमेंट के लिए किया जाता है। और इसके साथ में मैं यहाँ पे आपको कुछ ऐसे रेफरेंस भी दिखा रहा हूँ कि ऑलरेडी इस पर्टिकुलर प्रॉपर्टी के ऊपर कहीं ना कहीं कुछ ना कुछ काम हो चुका है जो कि इस पर्टिकुलर प्लांट की जो ये मेडिसिनल प्रॉपर्टी उसको प्रूव करते हैं। And the second plant is Garcinia pedunculata, that is known as Amlabetas, and it is known as Gulmaketu in Ayurveda. But this means ये पर्टिकुलर प्लांट में एंटी ट्यूमर एक्टिविटी है और ये ट्यूमर के लिए ट्रीटमेंट में प्रयोग किया जाता है। एंड सेम थिंग इज अजसरिंगी, मीन्स अजस्रिंगी जिम्नेमा सिल्वेस्ट्रिस, इसको बोटेनिकली जानते हैं। इसकी दो मेन प्रॉपर्टी, द फर्स्ट वन इज चक्षुष्या एंड अनदर इज प्रेमेहाग्नि। हियर द मीन्स ऑफ चक्षुष्या दैट इज ऑप्थेल्मिक, मतलब गुड फॉर आई, एंड प्रेमेहाग्नि मतलब पर्टिकुलर ऐसा प्लांट जो डायबिटीज की चिकित्सा के लिए प्रयोग किया जाता है। और यहाँ पे जो बहुत सारे रेफरेंसेस हैं वो इस बात को प्रूव करते हैं कि ये पर्टिकुलर प्लांट में ऑप्थेल्मिक एक्टिविटी है और डायबिटीज की चिकित्सा के लिए इसका प्रयोग भी किया जा रहा है। एंड नाउ अपराजिता क्लैटोरिया टर्नेसिया, इसको बोलते हैं विषहंत्री। इस पर्टिकुलर प्लांट का प्रयोग टॉक्सिन की चिकित्सा के लिए किया जाता है, या फिर जितने भी तरह के पॉइजनिंग होती है इनके ट्रीटमेंट के लिए इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। एंड वेरी लॉन्ग जो टीनोस्पोरा कॉर्डिफोलिया है उसके विषय में आप सभी बहुत अच्छी तरह से जानते हैं। पूज्य स्वामी जी महाराज हमेशा इसको ज्वर या अन्य बीमारियों की चिकित्सा के लिए करते हैं। तो इसका एक बहुत ही यूनिक और फेमस संस्कृत नेम है, डेट इज ज्वरारी। इट मीन्स जो ऐसा पर्टिकुलर प्लांट जो फीवर के ट्रीटमेंट के लिए प्रयोग किया जा रहा है। एंड सेम थिंग इज भल्ला तक फॉर द क्रिमिग्ना। कृमि का मतलब हो गया वम्स, और इस पर्टिकुलर प्लांट का प्रयोग जो है वो कृमि रोग की चिकित्सा के लिए किया जा रहा है। एंड नाउ ज्योतिषमती सिलेस्ट्रस पेनिकुलेटस, ज्योतिषमती का एक प्रॉपर्टी है, इसको बोलते हैं मेथ्या। इट मीन्स गुड फॉर ब्रेन। ये पर्टिकुलर प्लांट मेमोरी इनहेंसिंग एक्टिविटी के रूप में प्रयोग किया जाता है। और बहुत सारे ऐसे साइंटिफिक या बहुत सारे ऐसे एंशियंट फॉर्मुलेशन है जिस पर्टिकुलर फॉर्मुलेशन में इस पर्टिकुलर प्लांट का प्रयोग किसी न किसी ब्रेन रिलेटेड डिसऑर्डर या मेमोरी रिलेटेड डिसऑर्डर के लिए किया गया है। नाउ बॉसवेलिया सेराटा, इसको सल्लकी भी बोलते हैं, और ये इसमें कार्डियो प्रोडक्टिव एक्टिविटी पाई जाती है जिसको आयुर्वेदा में हृदय नाम से जानते हैं। एंड कैरम करबी मीन्स जीरक, इसको बोलते हैं दंत शोधनी। जितने भी डेंटल रिलेटेड डिसऑर्डर होते हैं उस पर्टिकुलर प्लांट, ये पर्टिकुलर प्लांट उस दंत रिलेटेड डिसऑर्डर के लिए या उनकी चिकित्सा के लिए इसका प्रयोग किया जाता है। नाउ कंद, सभी लोग इससे परिचित, इसको लोकल लैंग्वेज में सूरण भी बोलते हैं। बोटेनिकली इट इज नोन एज एमोर्फोफिलस कैम्पेनुलेटस, और इट इज अ वेरी गुड मेडिसिन फॉर द हीमोरॉइड्स। आचार्यों ने बहुत ही क्लासिकल बेस इसको डिस्क्राइब किया गया है। अगर हम हीमोरॉइड्स की बात करते हैं, उसी कंडीशन की बात करते हैं, चाहे वो 3 o'clock डिग्री पे हो, 6 o'clock डिग्री में हो या 9 o'clock डिग्री में हो, चाहे वो ब्लड पाइल्स हो या फिर हम बोलते हैं कि किसी भी कंडीशन की अवस्था का अगर हीमोरॉइड्स हो, उस पूरी तरह के हीमोरॉइड्स के कंडीशन के लिए इट इज अ वेरी गुड मेडिसिन। एंड सेकंड इज इन्युलर एसिमोसाइट इज नोन एज पुष्कर मूल, एंड द ड्रग इज यूज्ड फॉर द ट्रीटमेंट ऑफ द रेस्पिरेटरी ट्रैक्ट डिसऑर्डर्स, इसलिए इसका एक नाम है स्वासारी। एंड ट्रैकिस्पर्मम अमी, इसको यवानी या सूलहंत्री के नाम से जानते हैं। तो जितने भी तरह के पेन होते हैं उन सभी के ट्रीटमेंट के लिए इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। एंड दीज आर द सम रेफरेंसेस। नाउ अगर हम बात करते हैं, अभी तक तो जो मैंने बात की वो एंशियंट क्लासिफिकेशन में या एंशियंट सिस्टम में जो भी बहुत सारे टर्म्स ऐसे थे जिनको यूज किया गया था, इथनोफार्मोलॉजिकल के जो टर्म्स थे जिनको हम यूज कर रहे थे किसी न किसी ट्रीटमेंट के। अब यहाँ पे मैं बात करता हूँ कुछ ऐसे प्लांट्स हैं जो हमारे बॉडी के ऑर्गन से मैच करते हैं, और पर्टिकुलर उन प्लांट्स का प्रयोग सेम बॉडी ऑर्गन के ट्रीटमेंट के लिए किया जाता है। नाउ द फर्स्ट इज जुगलन्स रेजिया, इट इज नोन एज इन आयुर्वेदा एज अ करपलार और अक्षोट। ये पर्टिकुलर प्लांट बिल्कुल ब्रेन के जैसा दिखता है और इसका प्रयोग ब्रेन से रिलेटेड डिसऑर्डर के लिए या ब्रेन सेल्स रिलेटेड डिसऑर्डर के लिए किया जाता है। एंड सेम थिंग इज गुडुची मीन्स ट्यूनेस्पोरा कॉर्डिफोलिया। ये इस पर्टिकुलर प्लांट का प्रयोग, क्योंकि आयुर्वेद में डिस्क्रिप्शन दिया गया जितने भी क्रॉनिक डिसऑर्डर्स होते हैं उन क्रॉनिक डिसऑर्डर्स के ट्रीटमेंट के लिए गुडची का प्रयोग किया जाता है। एंड ऑल क्रॉनिक डिसऑर्डर्स रिलेटेड विद लीवर, तो हमें जो गुडची का प्रयोग है वो बिल्कुल लीवर की तरह दिखाई देता है। तो जितने भी लीवर रिलेटेड डिसऑर्डर्स होते हैं उन सभी डिसऑर्डर्स के ट्रीटमेंट के लिए टीनोस्पोरा कॉर्डिफोलिया का प्रयोग किया जाता है। एंड नेक्स्ट इज सिसस क्वाड्रेंगुलरिस, जिसको कि अस्थि श्रृंखला या हड़जोड़ के नाम से जानते हैं। बिल्कुल ये बोन्स की तरह दिखाई देता है और स्केलेटल सिस्टम की तरह दिखाई देता है, और इस पर्टिकुलर प्लांट का प्रयोग हमारे बोन्स से रिलेटेड डिसऑर्डर के ट्रीटमेंट के लिए किया जाता है। एंड नेक्स्ट इज ट्राइबुलस टेरेस्ट्रिस, जिसको हम गोक्षुर बोलते हैं। गोक्षुर एक वेल नोन प्लांट है जिसका प्रयोग किडनी रिलेटेड डिसऑर्डर के लिए और स्पेशली खासतौर से हम बात करते हैं यकृत आस्मरी और जो है हमारी ब्रिक्रास्मरी या रीनल कैलकुलस के ट्रीटमेंट के लिए इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। एंड नाउ एस्परेगस रैसिमोसस, जिसको सताबरी, ब्रिस्या, पीबरी, स्तन्यवर्धिका, ये सारे नाम इसको बोलते हैं, और लैक्टेशन प्रमोट करने के लिए इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। और नेक्स्ट है करंज, जिसको बोलते हैं कृतमाला। इस पर्टिकुलर डिसऑर्डर्स का प्रयोग स्पर्मेटोजेनिक की तरह किया जा रहा है। बहुत सारे ऐसे प्रोडक्ट आज भी या बहुत सारे ऐसे फॉर्मुलेशन हमारी एंशिएंट आयुर्वेदा में अवेलेबल हैं जिन फॉर्मुलेशन में करंज का प्रयोग स्पर्मेटोजेनिक एक्टिविटी के लिए किया गया है। नाउ सेंटेला एशियाटिका, इसका एक संस्कृत नाम मेध्या भी है, और इसका प्रयोग मेमोरी इनहेंसिंग ड्रग के रूप में किया जाता है सभी जगह। फिर है एवेलमोस्कस एस्कुलेंटस, जिसको बोलते हैं भेंडा या भिंडी, सभी इससे परिचित हैं। तो लेगर एंड सेल्स होते हैं, इसका जो टीएस काटते हैं बिल्कुल वैसा ही दिखाई देता है, सिमिलरली दिखाई देता है, और इस पर्टिकुलर प्लांट का प्रयोग डायबिटीज से रिलेटेड ट्रीटमेंट के लिए किया जाता है। नाव बाहुनिया वेरीगाटा, इट इज अ वेरी वेल नोन प्लांट, और इसको कांचनार बोलते हैं, और इसका प्रयोग जितने भी थायराइड रिलेटेड डिसऑर्डर होते हैं उन पर्टिकुलर डिसऑर्डर्स के ट्रीटमेंट के लिए किया जाता है। एंड मोमोटिका चेरेंसिया, संस्कृत में इसको कार्बिलक बोलते हैं, आप सभी जानते हैं इसे बहुत अच्छी तरह से। तो जितने भी पैंक्रियास से रिलेटेड डिसऑर्डर्स होते हैं उन सभी डिसऑर्डर्स के ट्रीटमेंट के लिए इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। फिर है सोलेनम लाइकोपर्सिकम, जिसको टमाटर बोलते हैं। तो टमाटर के ऊपर बहुत सारी रिसर्च एक्टिविटीज हुई हैं और इस पर्टिकुलर ये अगर हम बात करें ये क्लासिकली प्लांट नहीं है, एंशियंट जो हमारे ट्रीटेज हैं, एंशियंट आयुर्वेदिक जो ट्रीटेज है उनमें इस पर्टिकुलर प्लांट का डिस्क्रिप्शन नहीं पाया जाता है। फिर भी इस पर्टिकुलर प्लांट का प्रयोग बहुत तरह के कार्डियक रिलेटेड डिसऑर्डर्स के लिए किया जाता है। एंड डेकस करोटा आप सभी से बहुत अच्छी तरह परिचित हैं, इसको गाजर बोलते हैं और दैट इज वेरी गुड फॉर द आई। और अगर इसका हम रिजेंबलेंस देखें, अगर इसका टीएस देखते हैं तो बिल्कुल आई की तरह दिखाई देता है और इसका प्रयोग भी हमारा आई रिलेटेड डिसऑर्डर के लिए किया जाता है। एंड बॉम्बैक्स सीवा, जिसको सेमल बोलते हैं, सारे लोग एक्ने से बहुत अच्छी तरह परिचित हैं, तो इसका टॉपिकल एप्लीकेशन एक्ने की चिकित्सा में किया जाता है और इसका रिजेंबलेंस की बात भी करते हैं तो बिल्कुल ये एक्ने की तरह दिखाई देता है। एंड नाउ कस्कुटा रिफ्लेक्सा, जिसको आकाशवल्ली बोलते हैं, इस पर्टिकुलर प्लांट का प्रयोग हेयर से रिलेटेड डिसऑर्डर के लिए, क्योंकि इसमें ट्राइकोजेनिक एक्टिविटी होती है, तो हेयर से रिलेटेड डिसऑर्डर में इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। एंड फाइकस रेसिमोसा, इसको उदुम्बर बोलते हैं, इसका प्रयोग स्पर्म रिलेटेड डिसऑर्डर के लिए किया जाता है और एंशियंट क्लासिक्स में इसका बहुत ही अच्छा डिस्क्रिप्शन मिलता है। इसके अलावा एक प्लांट है म्यूकना प्रुडेंस, जिसको क्रॉन्च बोलते हैं, जिसको कि स्पर्मेटोजेनिक एक्टिविटी के लिए यह एफरोडिसिक माना जाता है और जितने भी डिसऑर्डर्स होते हैं, चाहे वो मेल रिप्रोडक्टिव सिस्टम का डिसऑर्डर हो या कोई और डिसऑर्डर हो, उन सभी में इस पर्टिकुलर प्लांट का प्रयोग किया जाता है। एंड नाउ हेलेक्ट्रिस आइजोरा, इट इज अ वेरी नोन ड्रग इन आयुर्वेदा, इट इज नोन एज आवर्तकी। तो अभी क्या है कि मैक्सिमम बहुत सारे लोग एंटी स्वाजोटिक ड्रग के रूप में डाइसाइक्लोमिन का प्रयोग करते हैं, बट इज द बेस्ट मेडिसिन फॉर द एब्डोमिनल पेन। ऐसा बहुत सारे रिसर्चेस इसमें और बहुत सारे क्लासिकल एविडेंस ऐसे मिलते हैं कि आवर्तनी का प्रयोग जो है वो उदर सूल की चिकित्सा में किया जाता है। एंड नाउ डाडिम, रक्त बीज जिसको बोलते हैं, रक्त का मतलब ब्लड भी, अगर इसके हम एक संस्कृत के हिसाब से अगर इसको डिस्क्राइब करते हैं या संस्कृत हिसाब से इसका मीन्स बताते हैं तो हियर द रक्त मीन्स ब्लड। इसके जो सीड्स होते हैं वो हिमेटेनिक होते हैं, वो रक्त को बढ़ाने के काम में आते हैं। ऐसा बहुत जगह पे क्लासिकल रेफरेंसेस मिलते हैं। ना ओलिया यूरोपिया, ये भी एक ऐसा प्लांट है जिसका डिस्क्रिप्शन एंशियंट आयुर्वेदा टेक्स्ट में नहीं मिलता है, बट पर्टिकुलर प्लांट यूज फॉर दी ट्रीटमेंट ऑफ द ओवरियन डिसऑर्डर। इसका अगर सेब की बात करते हैं तो सेब बिल्कुल ओवरी की तरह दिखाई देता है और ओवरी के डिसऑर्डर में इसका प्रयोग किया जाता है। एंड कस्कुटा रिफ्लेक्सा, अगर हम बात करते हैं तो जिस तरह से मॉडर्न में लैक्जेटिव, कैथार्टिक, इस तरह की बहुत सारी प्रॉपर्टीज बताई गई हैं, सेम कंडीशन हमारे आयुर्वेद में भी डिस्क्राइब की गई है, मृदु विरेचक, सूक्ष्म विरेचक और तीक्ष्ण विरेचक के रूप में। तो जितने भी जीआईटी रिलेटेड डिसऑर्डर होते हैं, जितने भी इंटेस्टाइन के डिसऑर्डर्स होते हैं, उन सभी में जो अमलतास है उसका बहुत ही अच्छा प्रयोग है, क्योंकि ये माइल्ड लैक्जेटिव की तरह काम करता है। ये कुछ इसके इंपॉर्टेंट रेफरेंसेस है जो इस पर्टिकुलर चीज़ों की पुष्टि करते हैं। एंड नाउ इसी चीज को ध्यान में रख करके जो हमारा ये एंशिएंट विजडम है, जिसमें बहुत सारे इथनोफार्माकोलॉजिकल एविडेंसेस मिलते हैं, चाहे वो एक साइन के रूप में मिलते हो, चाहे वो किसी न किसी प्रॉपर्टीज के रूप में मिलते हो, चाहे वो किसी न किसी नॉमेनक्लेचर के लिए प्रयोग किए गए हैं, इसी को आधार बना करके एक पर्टिकुलर प्लांट ग्रुप है जिसको हम अष्टवर्ग बोलते हैं। अष्टवर्ग में एक प्लांट है जिसको हम लोग काकोली बोलते हैं। उस काकोली के ऊपर एक इन विवो स्टडी प्लान की गई इसी पर्टिकुलर चीजों को बेस बना करके। तो अगर हम बात करते हैं, देयर आर लॉट ऑफ कंट्रोवर्सी इन अष्टवर्ग। अष्टवर्ग मीन्स ए ग्रुप ऑफ 8 मेडिसिनल प्लांट, दैट इज काकोली, क्षीर काकोली, जीवक, ऋषभक, मेधा, महामेधा, रिद्धि एंड वृद्धि। इस 8 में से एक पर्टिकुलर प्लांट है जिसको हम लोग बोलते हैं काकोली, और इस काकोली के नाम से बहुत सारी कंट्रोवर्सी एग्जिस्ट करती है। पर सारी कंट्रोवर्सी को रिजॉल्व आउट करने के बाद लास्ट में ये मिला कि 3 ऐसे प्लांट हैं जिनका प्रयोग काकोली के नाम से किया जाता था। फर्स्ट वन इज रॉस्कुइया एल्पिना, सेकंड इज कैपिटाटा एंड थर्ड वन इज प्रोशेरा। इन तीनों प्लांट को बेस बना करके, और यही जो इथनोफार्माकोलॉजिकल अप्रोच है, इस अप्रोच को बेस बना करके, या जो क्लासिकल एविडेंसेस मिलते थे कि ये पर्टिकुलर प्लांट एक्ट एज अ स्पर्मेटोजेनिक, उस पर्टिकुलर इथनोफार्माकोलॉजिकल एविडेंस को मान करके इस प्लांट के ऊपर आगे काम किया गया, और इसमें स्पर्मेटोजेनिक एक्टिविटी और सेक्सुअल बिहेवियर स्टडी देखी गई। और इसमें जो एडॉप्टेड मेथड है, इसका हम अगर एक्यूट टॉक्सिसिटी के बाद में, क्रोनिक टॉक्सिसिटी के बाद में इसकी एनिमल स्टडी हुई, तो लास्ट में क्या सजेशन आता है, लास्ट में जो आउटपुट आता है वो आता है कि जो रॉस्कुइया एल्पिना है, रॉस्कुइया एल्पिना बहुत ही सिग्निफिकेंट इंक्रीज और कॉपुलेटरी एफिशिएंसी और स्पर्मेटोजेनिक एक्टिविटी को शो करता है। इट मीन्स ये है कि हम जो बहुत सारे क्लासिकल एविडेंसेस हैं हमारे पास में, ये बहुत सारे जो इथनोफार्माकोलॉजिकल एविडेंसेस ऑलरेडी डिस्क्राइब इन अवर एंशिएंट टेक्स्ट, उनको बेस बना करके बहुत सारी ऐसी कंट्रोवर्सी है जिनको रिजॉल्व भी कर सकते हैं, और बहुत सारे ऐसे फील्ड है जहां पे हम बहुत ही यूनिक काम कर सकते हैं। लास्ट में मैं ये कहना चाहूँगा कि जो नेचर है हमारा, वो ह्यूमन एक रिजर्वायर है ह्यूमन वेलबींग का, और बहुत सारे सीक्रेट्स है उसमें, चाहे वो उसके ट्रीटमेंट के लेकर के हो, चाहे वो उसके प्रिवेंशन के लेकर के हो। तो हमें फिर से इस पर्टिकुलर एंशिएंट नॉलेज के ऊपर एक बार विचार करके, या बहुत सारे जो इथनो फार्माकोलॉजिकल साइन और सिम्टम है, या जो भी इसके पर्टिकुलर सिग्नेचर दिए हैं, उनको विचार करके फिर इस दिशा के ऊपर एक बार और काम करना चाहिए। और पतंजलि ऑलरेडी इस दिशा में आदरणीय डॉक्टर अनुराग वार्ष्णेय सर के निर्देशन में बहुत सारे काम कर रहा है, जिसका ही परिणाम है कि आज बहुत सारे जो ग्रिट फॉर्म की जो मेडिसिन है, वो कहीं ना कहीं इस बात की पुष्टि करती है कि द टोटल मेडिसिन आर डिपेंड ऑन द इथनोफार्माकोलॉजिकल साइंसेस। थैंक यू।
Professor Yuan-Xian Chang to deliver his talk on quality control of TCM herbs and herbal preparations in Taiwan. Dr. Cheng is professor at pharmacognosy at College of Chinese Medicine, China Medical University, Taichung, Taiwan. Professor Cheng is also appointed as honorary professor of Hong Kong Baptist University. To date, he has published 160 scientific papers and 27 books in TCM-related fields. It's a pleasure to have you here, sir. Let's all give him a very warm welcome.
Okay, yeah. Thank you for the great applause. Good afternoon. First, I'd like to thank the organization committee for inviting me to participate in such a great event. And particularly, I'd like to thank the Professor Bhajan, the Vice President of the Institute. In India, the traditional medicine, yoga, and also Ayurveda. But in Taiwan, we have Chinese community. We have a traditional Chinese medicine. So today, I'd like to share with you our traditional Chinese medicine. And I personally, I'm the pharmacist. I'm not a TCM physician. So I'd like to share with you on the quality control of TCM herb and herbal preparations in Taiwan. As you understand, traditional Chinese medicine has been very popular in Taiwan in the past, and TCM was also incorporated into our national health insurance since 1995. So with the National Health Insurance ID, every individual in the nation, we can visit any clinic, either Western medicine, Chinese medicine, or even in the hospital. We just pay a very minimum amount of registration fee, and the rest are all covered by the insurance. And of course, we pay the insurance fee based on the income of the individuals. And of course, the quality control of TCM herb is very important. So in my talk, I'm going to talk about some regulation on the quality control practice in TCM, together with some of the work we did, particularly Taiwan Herbal Pharmacopoeia. And today I bring one copy of the Taiwan Herbal Pharmacopoeia, the newest fourth edition. Very likely tomorrow I'll bring as a souvenir to the Institute. As you understand, traditionally we have a TCM herb. And when the patient brings, after making diagnosis, the TCM physician may draw the prescription. Prescription goes to my department. I used to be the head of a herbal pharmacy. And then as the patient brings all the herb home, they have to make tea, make decoctions. Of course, it's time consuming. But nowadays, the more popular dosage form is extract granule. By taking 4, 5, or 6 grams of the extract granule powder, you will be equivalent to drinking a cup of herbal tea. It's more convenient. And of course, those extract granule, we have extract granule from a single herb, or extract granule from a compound formula. And those are the product of a TCM pharmaceutical company in Taiwan. So this is my outline. As you understand, the quality, the identity of the herb is very important. If you don't use the correct species, definitely it will not be able to achieve expected clinical efficacy, may even cause poisoning. So usually we have 6 common identification methods: species identification, morphological, microscopic, chemical, biological, even most novel DNA analysis. When we walk into the field, particularly I'm going to visit your herbal garden tomorrow, hopefully. When we walk into the field, by taking a look of the herb, the tree, whether it's a woody or herbaceous, taking a look of the leaf, of the flower, we try to identify the species. And sure, it's not easy even for expert. But however, with the crude herb, we only take part of the plant. For example, with this, without the leaf, without the flower, what is it? Therefore, the identification of the crude herb are far much more difficult than that of the plant. Of course, for the most commonly used TCM, at about 200 or 300 TCM herb, things are much easier. And in academic institute, we also do microscopic identification. We try to do cross-section, observe under the microscope. We also observe the powder features. And however, I'd like to point out that, I apologize for I think the equation just moved away. I'd like to point out, just doing microscopic identification means nothing. For example, with the crude herb, we don't know what it is, right? Then why don't you do cross-section? Even after you do the cross, you will not still be able to tell you what is it. Therefore, the reference standard or reference are very essential to compare with. And of course, nowadays, we have the senior chromatography TLC identification. And for example, from temperature sample, the marker ingredient can be observed from all temperature samples. And of course, nowadays, we have the quantitative analysis. We use HPLC, UPLC, or even GC. And this fingerprint from temperature sample. We also do biological evaluations and most DNA identification, etc. But however, in pharmaceutical company, besides those general identification methods, they also do some chemical specification examination, such as loss on drying, total ash, insoluble ash, etc. We usually call it boring routine. Those work were always assigned to the beginner to do those most basic work. But however, a couple of years ago, we encountered so-called weight gaining powder. What they did was they dissolved this white powder into the hot water as much as possible until saturated. And after that, they dip the herb into the solution for some while, and then take it out and dry it up. And it's going to increase the weight of the herb 10%, 20%, or even up to 30%. And for the herb of light nature, they even increase weight up to 100%, 200%. Of course, it took us some while to figure out it was magnesium sulfate. It's very cheap chemicals. Of course, it's not good. But however, if you do those boiling routine regularly, the batch of sample that was adulterated with so-called weight-gaining powder magnesium sulfate is going to end up with high total ash content. You can just reject this batch of sample just because of its with high total ash content. You don't need to tell, oh, you adulterate with magnesium sulfate, I'm going to reject this batch of sample. So even though those are boring routine, but it makes sense in some cases. And therefore, and also the drug standard is called pharmacopoeia. And as to the herb, we have herbal pharmacopoeia. The first herbal pharmacopoeia in Taiwan was published in 2004, with only 200 herb items. Definitely, it's not good enough. But however, with this first edition, we set the sulfur dioxide limit, heavy metal, pesticide residue, and even aristolochic acid cannot be detected. And we also do all those chemical specification background for the new item to be included in our new edition of Taiwan Herbal Pharmacopoeia. So for the second edition, the item increased from 200 to 300, and previously we have color reactions, and which was not specific. So it was replaced by TLC and HPLC. And we also incorporate some safety test limits such as pesticide residue, heavy metal, aflatoxin, etc. into each individual monograph. And we try to use the safer solvent, not to use the poisonous solvent. and we also respect the industrial experience. And in the past, the detection method was less in the appendix with page number, but later on it was replaced by a 4-digit number for each detection method. And we also, we call Taiwan Herbal Pharmacopoeia and abbreviate as THP. And besides that, we also compiled the color illustration of Taiwan Herbal Pharmacopoeia. What we did was we provide the color photo of the plant, of the crude herb, and also with the decoction pieces. That's why I like to go to the botanical garden to take some additional photos of the plant. And also I'm the author, and Professor Dr. He was also co-author of this book, and he was also provided with the CD-ROM. And this book received the first publication award from our Ministry of Health and Welfare. And for international exchange, we also translate our Chinese herbal pharmacopoeia, Taiwan herbal, into English. And the newest edition was the 4th edition, which I'm going to send it to your institute tomorrow. And I'm the PI, and Dr. He is the co-PI. And based on our work, the government later on published official copy of Taiwan Herbal Pharmacopoeia based on our draft version. And as to the 3rd edition and 4th edition of Taiwan Herbal Pharmacopoeia, in order to invite more experts to join the editorial committee of the Taiwan Herbal Pharmacopoeia, 4 subcommittee was set up, like Source Working Party, Assay Working Party, Preparation Working Party, and Clinical Chinese Medicine Working Party. And this is the key editorial board members. And so currently we have the 4th edition of Taiwan Herbal Pharmacopoeia, both English and Chinese version. And it all can be downloaded for free from the website. And on the 3rd edition, the herb item increased from 300 up to 350 herb items. And besides that, we also have 2 extract granule preparation, one compound formula, the other a single herb extract granule. And we also set some sulfur dioxide limit for 25 most frequently fumigated TCM herb with 400 ppm. And the rest should not exceed 150 ppm. And we also have the general limit for aflatoxin for the 37 TCM herb. And then the limit was 10 ppb, while the B1 are more poisonous, more toxic, so the B1 not more than 5 ppb. And we also have a general limit for heavy metal. So this is the 2nd edition and the 3rd edition comparisons. And we also have some workshop during the time period, and many international experts such as Professor Ludi Bauer, Francis from Germany, and Professor Kota from Japan, and Professor Robert Ma from Canada were invited to participate in those workshops. So as I told you, the Taiwan Herbal Pharmacopoeia is available for free downloading from the website. The philosophy behind that was, this is regulation. As more people know the lure and then follow the lure, it's better, rather than sell it at a very high price and then try to make money from there. It doesn't make sense. And as to the 4th edition of Taiwan Herbal Pharmacopoeia, the origin source, they try to, on this version, they changed the Latin name of the plant species dramatically, following the MPNS from the Kew Botanical Gardens. For example, Eugenia caryophyllata, the name I used to know for some 40, 50 years, but it was changed to Syzygium aromaticum. But however, through my suggestion, we put the in parentheses that old traditional name in tradition, so for people like me can understand what is it. But however, in CP 2020 in China, they still use the old traditional Latin name system. And personally, as to the Assay Working Party, currently I'm the chair, the chairperson for this subcommittee, and Dr. He is the PI for this session. And we also provide the calculation equations for easy calculations. And as to the preparation working party, on this version, a total of 9 TCM preparation monograph was incorporated. And as to the clinical physician working party, it's unique because in most of the other herbal pharmacopoeia, they never invited clinical physician to join the job. But however, with almost about 15 TCM physicians' participation, it seems they pay more attention to the property and flavor, tropism, usage, dose, precaution, or even poisonous TCM herb item. So this, the first edition was published in 2021 with new herb item. And again, it's all both Chinese and English version were available for downloading from the website. Actually, you don't need to remember the name of the website. You just key in Google, Taiwan Herbal Pharmacopoeia. You are going to get whatever you need for free downloading. And besides that, currently we have 80 GMP pharmaceutical company, and since 2005, all the TCM pharmaceutical company has to meet GMP standard. And most of the pharmaceutical companies located in the southern part of Taiwan. And currently, since 2020, the government start moving one step forward up to a CGMP. And hopefully by 2026, all pharmaceutical companies in Taiwan will be able to meet this CGMP standard. And lastly, in 2 or 3 minutes, I'd like to share with you, my team was also invited by the Department of Health, Hong Kong, to join the so-called Hong Kong Chinese Material Medical Standard. The term may sound strange to you, but actually it can be called Hong Kong Herbal Pharmacopoeia. By pharmacopoeia referring to the drug standard of a country. Hong Kong is not normal, it's just the local government. They should use the Chinese pharmacopoeia. But however, since they are currently still use Hong Kong dollar rather than Chinese dollar, okay? So they are two different systems. So they try to have their own. And they returned back to China in 1997, but 5 years after that, in 2002, they start the HKCMS project. And my university, the Minister of Health of Hong Kong visited my university and invited my team to join the job. So for those who are interested, it's available online, both Chinese and English version, okay? And it had been 7 contracts had been signed between the university and also the Department of Health. They signed the contract with the president of the university, and I'm just a PI who carry the research work. And those are some photos. They visit my university. And then also the Secretary of Food and Health Bureau of Hong Kong also visit my university. And currently I'm the PI and Dr. He is a co-PI. So every 2 or 3 months we have to fly to Hong Kong to attend the regular scientific meeting. But however, in the last 3 years, due to the COVID, many meeting was postponed. And lastly, I also invited to participate in the European Pharmacopoeia, ETQM TCM Working Party, and also the USP. Currently, the Ludi Bauer from Austria is the chairperson for this TCM Working Party. And this is the photo we took a couple of years ago. And 3 meetings were held each year. And however, in the last 2 or 3 years, most of the meetings were held in virtual mode. And this is the virtual mode. And besides that, I also invited to participate in the USP Herbal Medicine Compendium East Asia Expert Panel. Currently, the panel was chaired by Professor Guodean from Shanghai. And that's it. And 2 years ago, I was invited one step upward by the USP as USP Botanical Dietary Supplement and Herbal Medicine Expert Committee. And currently, the committee was chaired by Professor Robert Mao from Health Canada. And actually, this Robert Mao, actually, he is my classmate in the PhD program in University of Illinois at Chicago in some early days. And I also still keep as member as the Far East Expert Panel member. And personally, I was also invited in the headquarters of the USP, and just last week, 2 days before I leave for this meeting, the USP, we are going to have another USP meeting in USP headquarters by the end of the March. So in conclusion, I'd like to share with you that participants in Hong Kong Standard Project, USP, European Pharmacopoeia, keep aware of the current international trend and on the quality control of TCM, and we can also share Taiwanese experience. And the cooperation on the herbal pharmacopoeia between the country can be strengthened. And the chemical background information and the development detection method should be shared. And each country can then make their own remedy based on their country need. And for example, many herbs you use in Ayurveda medicine are common to what we use in China. So we can share this kind of information. Thank you so very much. Thank you. Okay, good. Bye-bye.
Thank you, sir, for sharing your valuable insights on quality control of TCM herbs and herbal preparations in Taiwan. Moving ahead, now I'd like to invite the next eminent speaker for this session, Dr. Sandeep B. Bharate ji, for the traditional medicine in India. Dr. Bharate is a senior principal scientist and head of the department at Natural Products and Medicinal Chemistry Division, CSIR. He is also an honorary professor at Academy of Scientific and Innovative Research, India. His laboratory focuses on the natural products-driven drug discovery of new chemical entities and phytopharmaceuticals. It is a privilege to have you here, sir.
Okay. Very good afternoon. Okay. So before I start my talk, I must thank Dr. Vashne Acharya ji for this my wonderful opportunity to talk about phytopharmaceuticals, where we, as a CSIR IIIM, we have been working in this area since last 4 to 5 years. So before I start speaking about the phytopharmaceuticals, the institute from where I'm coming is the CSR laboratory. One of the laboratory of the 38 CSR labs are there in India. Out of that, this is the laboratory which is located extreme towards the north. And this laboratory was established even before establishment of the CSIR, even before independence of India. So this lab was established in 1941, and after that, 1942, CSIR was started. Until 1957, it was under the Jammu and Kashmir government, and later on, it was taken by the CSIR with the name of Regional Research Laboratory. And after almost 50 years, it was realized the mandate of the institute that integrating the traditional knowledge with the modern biology. So based upon that mandate, the name of the institute was changed as Indian Institute of Integrative Medicine. So since those almost 70 years are completed, and this lab was very well known for isolation of the bulk natural product. All these natural products, they used to get exported to the European countries. So before I start speaking about, so I work in the area of natural products, and the main focus of our research is focus at the natural product scaffolds. So we work in the area of 3 areas, like 3 to 4 areas. Phytochemical investigation of some of the interesting medicinal plants. Then once we isolate some natural product, we pursue its medicinal chemistry. These are the 2 therapeutic areas, oncology and CNS diseases. And parallelly, we also focus on identification of some phytopharmaceutical leads and nutraceutical products. And in both the areas, once something comes out which is has a potential for a preclinical validation, we go to the preclinical studies as per the regulatory guidelines of the DCGI, and so that we can take something to the clinical trial. So some of the 2 case studies I will present it here. So these are the contents of my talk. So before I speak about our own case studies, I will show you the briefly speak about the regulation of traditional medicine global. What is the global status? So even before speaking about the India, I will speak about the US FDA guidelines, European Medical Agency guidelines, and CDSCO guidelines for herbal medicines. And then what are the opportunities, what India has done, what CSR has taken the initiatives. And there are 2 case studies which I will present how we are working in the area of phytopharmaceuticals. So globally, if you see herbal medicine, I think since morning we have been listening that this is very important alternative to the modern allopathic medicine. And there is no doubt about that. So, but there is a reason why still medicinal practitioners, these MBBS doctors, why they are not prescribing the herbal medicine? Because there is a lack of scientific validation. Everyone knows that. And still we have to promote the herbal medicine. The reason stands here like there is no scientific validation, and there is where the phytopharmaceutical guidelines and these botanical guidelines have been created. In United States, there is an example I'm mentioning here, like there are 50,000 side effects, adverse effects were observed with some of the food dietary supplements and herbal medicines, and because of that reason, they created separate guidelines for the botanicals, and also very strict guidelines for the toxicology information, regulatory toxicology they have mentioned. So although they started taking IND applications from 1985 for botanicals, but actual guidelines were created in 2004. And very recently, they have been revised in 2015 with the addition of some of the more toxicological information. So understanding these USFDA guidelines and looking at the traditional wealth, Ayurvedic traditional knowledge available in India, Indian government came forward and they created separate guidelines under the CDSCO, and they created a guidelines with the name of phytopharmaceuticals. And this guideline came into existence in November 2015. So this is the global status of the regulation of the herbal medicine. So if you, from the picture itself, you can see that the blue portion where at least there are some sort of guidelines for regulation of herbal medicine, but the red dots mentioned here, you can clearly see that there are mainly in Africa, some of the countries in Africa, there are no any kind of guidelines for herbal medicine. And now this is the survey done by WHO in 1919 when they did the survey. So after 10 years, again they conducted another survey in 2012, and you can see the increase in the number of member states where there is a traditional policies there. Even there are regulations have been created, and there are the countries where they have created the separate research institutes for doing the research on traditional medicine. So many institutes, like India has several institutes, like there are a couple of traditional research institutes, like Patanjali, PRI is one of the institutes which is focused on the traditional medicine. Now I'll speak about the 3 important regulations. One is USFDA. So USFDA called it as a botanicals, and the guidelines were created in 2004. And in Europe, it is called as a herbal medicinal product, HMPs, and they created a guideline in 2008. Whereas India also created the same level of guidelines, same level of stringent guidelines in 2015. So in 2004, this USFDA created the guidelines, and until now, they have received 900 IND applications. This is a huge number. In last 18 years, they received 900 IND applications, and surprisingly, there is not a single IND application from India to the USFDA as a botanical. Though India claims that there is a huge traditional knowledge, Ayurvedic literature is there, and lot of, even in these 900 IND applications, many plants, 2/3 of the plant they are coming from the India. India is the origin for that. So those people are collecting the plants from India, doing research on that, and filing IND applications. So that means something is lacking in India. Indian scientists are not focusing on the scientific validation in a proper professional way. So this is, I think, major concern, and we should focus on this aspect. So USFDA, according to USFDA, well, what is botanical? So this definition, I'll just put it here. So according to USFDA, botanicals means any product which is coming from the plant materials, algae, macroscopic fungi, or combination. So they are open to plants, fungi, anything they are allowing. But it does not include the products that are coming from animals or animal parts, or any fermentation-driven products, or highly purified or chemically modified botanical substances. They do not fall under the botanicals. So this is the statistics of the IND applications to the USFDA under this botanical guideline. You can see the graph. 1984, slowly some of the INDs or even pre-INDs, companies started coming forward. And after getting the separate guidelines in 2004, see the rise in number. So per year now they are receiving more than 50 IND applications. This is a huge number. So this is the statistics, like out of 900 pre-INDs and INDs, there are 65% of the INDs are for the single herb, and 35% INDs are for the multiple herbs, and more is for the oncology. So what is the reason for oncology? Because from the inherent nature, plant, why synthesize secondary metabolites? Because for the self-defense mechanism, and that is the reason even whatever drugs are approved by the USFDA, single molecule allopathic drugs, almost 80% of the drugs are coming from the natural product pharmacophore. So main pharmacophore present in those anti-cancer drugs are natural products. And that is the reason there is a huge potential of the natural products or medicinal plants in the oncology domain. And out of these 900 INDs, there are only 2 reached to the clinical trial. And both have gone to the NDA, new drug NDA filing, and both have got FDA approval. So those examples I will show you. So this is the first FDA approved botanical drug in 2006, which is a Veregan, which is a mixture of catechins. It is called as cinecatechins, which is coming from the green tea extract, and it is a topical ointment for treatment of genital warts. And after 6 years, another US botanical approval was there in 2012. So this is again based upon the traditional medicine. There was in South America, there is a plant called as a croton lechery. So this plant, so this several parts or latex of the plant was used for treatment of diarrhea, fever. So there was a traditional knowledge was there. So one of the company, they came forward and they prepared a botanical extract and approached the US FDA. So US FDA, even without doing all animal pharmacology, just based on the traditional knowledge, they granted the permission to do the clinical trials. And they could complete the phase 2 and phase 3 clinical trial. And in 2012, they got the FDA approval as a first oral botanical drug for treatment, taking care of the diarrhea in HIV patients. So now after US FDA, I will speak about a little bit about what are the guidelines in European countries. So European countries also, they created a separate guideline for the botanical drugs, which is called as a herbal medicinal products. And they created 3 categories. So this is very important. One is a traditional use. So where there is a traditional use is already documented in traditional literature, you know there is no need of doing the animal pharmacology, toxicology, nothing is required. You can directly get the permission to do the clinical trial. Second category is the well-established use. At least if some medicinal plant is there or any product is there, which is being used for at least 10 years in the Europe, you can directly go to the clinical trial. You can get the permission from the EMA to do the clinical trial. And standalone means you have to establish each and everything, preclinical pharmacology, even ADME studies, regulatory toxicology, everything you have to do under the GLP guidelines. And then after doing all this, just like a small molecules, you have to do everything and then you can get a permission to the clinical trial. Now what about India? Now we'll come into the India. So India initially had only Ayush Ministry was regulating the herbal medicines. So which Ayush Ministry was created in 2006. And later on, after realizing this US FDA, EMA guidelines, Indian regulator came forward and they thought to create a separate guideline which are extremely stringent in terms of the toxicology information like US FDA. So that those MBBS or modern practitioners, they can prescribe this, the drug which is regulated by the CDSCO, they can prescribe this medicine. Whereas nowadays, Ayush medicines are not prescribed by MBBS doctors. So that is the reason there is a strict borderline is created between the Ayush products and CDSCO. So both these are differentiated by, there are some of the differentiating parameters like phytopharmaceuticals must be standardized with 4 chemical markers. There is a stringent requirement of the toxicology studies under GLP guideline, exactly like the small molecule or allopathic medicines. And in the same year, the FSSAI, there is another agency for the regulating the food supplements or dietary supplements. So they added one addendum in the 2016. So where functional foods can be developed and we can, on the label, you can make a claim of a therapeutic use. So because of these 2 important guidelines, like this is the schedule they added in the FSSAI guidelines, they created a list of 400 medicinal plants and the ratio also they mentioned, like for one of the plant, they clearly mentioned that this much quantity if you take, and if a product contains this much quantity of a particular plant, you don't have to prove any toxicology or safety information. You can directly make a product and launch in the market with the therapeutic claim on that. So this is another interesting factor here. So with the launch of these 2 important guidelines, CDSCO and FSSAI, this Schedule 4, so there is a huge opportunity for translating this traditional knowledge to the modern practitioners, modern drugs we can make out of these as a phytopharmaceuticals. So what is the definition of the phytopharmaceuticals? So it is exactly taken from these guidelines which is published in the November 2015. So it is defined as a purified or a standardized fraction. You have to go one more step beyond the extract. You have to make enrichment with the particular bioactive marker. So you must know what is your compound in the plant which is actually showing the biological activity. So it is a purified or standardized fraction with defined minimum 4 bioactive or phytochemical compounds. And these new guidelines are exactly in the line with the US FDA, Chinese and other European regulations. So what kind of information is required if you are doing all preclinical research and you want to file an IND application? So very briefly I summarize here. So first part of the IND application is that whatever knowledge is there in the literature, like published literature on the plant or a phytopharmaceutical drug, or any scientific reports are on there on the safety, pharmacological studies, or if there is a present usage, some products are already in the market. So those information you have to provide to the regulator. Then publish scientific literature on the pharmacological studies, even including the human studies, and if there are any monographs in the literature like Ayurvedic pharmacopoeia, that plant is documented. So all those information you have to mention in the IND dossier. Then second part comes is the data, what you generate as an applicant. So an applicant, like Dr. Chang has clearly mentioned, there is a requirement of identification, authentication, all these microscopy, macroscopy studies for the authentication of the plant material, raw material. And apart from this, very important is a chemical standardization. You have to standardize your plant material with the 4 chemical markers, and that is very, very important. And you have to specify the ranges, like when there is a reproducibility record, it's like one batch you make and after 5 years you make another batch, or you collect material from some other source. So to match those repeatability and reproducibility, you have to standardize the enriched fraction, and you have to specify the range. If your this batch falls within that range, only that batch will be passed for the production or commercialization. Then process of the extraction and subsequent fractionation and purification, all the details you have to provide in the IND dossier. What formulation you are making, because obviously it is an oral herbal drug, botanical drug means oral, whether it is a tablet, capsule or syrup formulation, all those details is required. Then manufacturing process of the formulation is required, and stability data under the two, this is normal long-term stability as well as accelerated stability conditions. So that's the data you have to provide. So while IND filing, you have to prove that your product is stable at least for 3 months, because clinical trial will happen for 3 to 4 months or maximum 6 months. So during that period, your product should be stable. So that data you have to provide initially, but ultimately you have to generate the data for at least 2 to 5 years. Then safety and pharmacological information, where all the GLP regulatory talks you have to provide, and that data has to be provided. And along with this, you have to provide the phase 1 protocol, because you are filing IND to get the permission for doing the phase 1 clinical trial. So that means you have to freeze the protocol, you have to submit that protocol, like how many patients will be there, or whether they will be healthy volunteers. All those information you have to provide as a part of IND dossiers. Now, briefly I will speak about what are the differences between the small molecule versus phytopharmaceutical. Small molecule means whatever allopathic medicine. So as far as the IND enabling or preclinical research is concerned, these are the 3 important, I think, pillars: CMC, regulatory toxicology, and ADME studies. So what are the differences between these small molecule versus phytopharmaceuticals? There is a broad difference in the CMC, like in small molecule, simply you check the purity or impurity profiling. Your compound is, small molecule is more than 99% pure, the particular impurities are less than 0.001%, so your molecule is okay to go ahead. And you have to do the pilot scale synthesis under the schedulium. Formulation also, any type is allowed, because in the small molecule it can be taken by intravenous route, intramuscular route, or any route of administration. But in phytopharmaceutical, these are the differences, like you have to do the chemical standardization with 4 markers, you have to check the repeatability, whether the same batch prepared during different times or plant collected from different location, different time of the year. So whether you are getting the same content, or at least within that range your marker compounds are coming. Since it is coming from the nature, you should check you have to specify that there are no aflatoxins, there are no pesticides, there are no heavy metals, there is no microbial load. All these parameters you have to provide. GMP manufacturing you have to do under the schedulium, although there are currently there are no facilities for schedulium for the herbal drugs, but our institute has created one facility for the GMP manufacturing, so where we can do the production of the GMP batches for the phytopharmaceutical leads. And obviously oral formulation, capsule, tablets or syrup. So these are the major differences between these two. As far as the regulatory safety is concerned, exactly similar toxicology information is required, like systemic toxicology, and in 2 species, one rodent and non-rodent. Both are required in small molecules and phytopharmaceuticals. Then reproductive toxicity, genotoxicity, cardiotoxicity, everything is required. As far as ADME concerned, in case of small molecule, you need very stringent requirement for the ADME study, like you have to do the PK, single dose PK, even dose dependent PK you have to check. Tissue distribution also you have to check, whether your compound is going to suppose your drug is targeting the lung cancer, whether compound is going to the lung or not. So that tissue distribution studies are required. Whereas in phytopharmaceutical, that much stringent requirement is not there. Only you have to make sure that your bioactive compound, or compound which is you are enriched for in that particular compound, particular phytopharmaceutical lead, that is going to the plasma or not, plasma circulation. So that confirmatory PK is required to make the sure that bioactive compound is reaching to the blood circulation. So now after looking at these guidelines, there are huge opportunities based upon the traditional knowledge available in India. A lot of Ayurvedic medicines are there, even Unani and Siddha medicine, where there also medicinal plants are there. So based upon this, there is a huge opportunity, there is a rich biodiversity, and there is a huge expertise also available in India. Only you have to put all these 4 pillars together so that we can do the scientific validation, and with this we can diversify towards the 2 directions. One is a phytopharmaceutical, if you are developing an enriched fraction, or you can identify a pure natural product and you can build a medicinal chemistry on that to come to the small molecule which can you go to the regulator as a small molecule or allopathic medicine. So that is a reverse pharmacology. So coming from the plant to the single molecule is a reverse pharmacology. So both the approaches are there, and for going to the phytopharmaceutical guideline, I must say that there is a huge robust CMC is very, very important. Unless and until there is a strong natural product chemistry, very strong feel about the pure natural product, you must build a confidence on the natural product. Like you are working on a plant, that particular, what is the compound which is actually responsible for the activity. So you must build a confidence that this is the natural product which is actually showing the activity. You must play around that natural product before shifting to the phytopharmaceuticals. So 2 examples I will show you from my own research. You will understand why I am saying this. So these are the 3 plants we have been working since last 4 to 5 years on this. So all these plants, I think at least 2 plants are particularly occur in the Jammu and Kashmir region. Bhajan silita occurs in the temperate Himalayas in Jammu and Kashmir. And this plant is documented in Ayurveda. It is called as a Pashana Bhet, and it is used in traditional literature. There are multiple products in the market for kidney stones, and even there are reports on the anti-inflammatory properties of that. Another plant is a Crocus sativus. You must be knowing saffron. So this is also used since ancient times for the memory boosting properties or so many properties. And third plant is a Diacetyl Banatriferum. This plant particularly present in the 2 places mainly, this Western Ghats and this FRI campus of the Dehradun. So this plant is present in that campus. And this so whenever we started working on these plants, we first identified what is the major bioactive compound which is actually showing the activity. So always we think about the natural product. We go one step beyond that. Like what are the natural products? Because we must build a confidence that natural product should be active. It must be stable, it is reaching to the plasma, it is going to the target organs, and if you test that pure compound in the biological assays, it is showing the activity. So that kind of confidence is required. So first plant where the Bhajan silita, this is the major compound bergenin, which is present in 1 to 2%. 1 to 2% in a plant is very good quantity. And so see, this is such a unique compound, is a phenolic glycoside, C-glycoside, and this compound we identified that it has interesting biological properties. It shows anti-inflammatory properties, as well as it reduces the pain. So there is a disease called as rheumatoid arthritis where there is a inflammation as well as the pain, and one compound, if it's taking care of the 2 major prong approach, like both the things, it is a wonderful approach. So on that approach, we started working on this. Another plant, Crocus sativus, it is a very rich source of the crocins, and this crocins actually we did all ADME studies, and that I'll speak about in my coming slides. And this Diacetyl Banatriferum, this is a unique plant which occurs only in India. And this natural product, if you see the structure, this has already given the 2 clinical candidates, and both are in the phase 2, phase 3 clinical trial for oncology. So such a plant which is giving the clinical trial, and from the IIIM also we discovered 1 candidate. I think I'm not speaking about that compound, IIIM 290, for which we got the DCGI permission to do the clinical trial for pancreatic cancer. So that is our one other medicinal chemistry we did on the natural product, and we discovered something for the pancreatic cancer. So first plant is a Bhajan silita. So this is documented in Ayurveda, and it is used for the kidney stones, and these are some of the products already in the market. So we started working on this plant, and first we isolated the pure compound from the plant. We collected the aerial parts of this plant, and this is the bergenin, and initially we wanted to play around this natural product. So we did medicinal chemistry on that. We synthesized almost 20 new compounds, semi-synthetic derivatives, and along with the bergenin and new compound, we tested for the in vitro in vivo assays of the inflammation and arthritis. Like it is inhibiting the IL-6 and TNF-alpha, but it is selective inhibitor of IL-6. So in rheumatoid arthritis, there are pro-inflammatory, there is increase in the levels of pro-inflammatory cytokines. And if you inhibit too much TNF-alpha, there is a chance that you get immunity goes down, and there are chances of infection. That is the reason whatever monoclonal antibodies are approved by the US FDA for the TNF-alpha, there is a black box warning on the label of the TNF-alpha antibodies, because there is a chance of getting a tuberculosis and HIV infection. And that is the reason if something is highly selective for IL-6 cytokine and not inhibiting TNF-alpha, that is a wonderful, small molecule. And here, that kind of molecule we identified, it is inhibiting IL-6 with the 200 nanomolar, and IC50 for TNF-alpha is 12 micromolar. Then we tested in the in vivo and in vitro models of the inflammation, arthritis also. We checked the cytokine levels. Even we checked the anti-immunogen antibodies also. So you can see this IgG1, IgG2A. So both these antibody levels also going down with this bergenin. we published this literature, and we also filed IND, patent on this pure compound and new semi-synthetic derivatives. Then we proceed for the phytopharmaceutical. After building a confidence on the natural product, it has an activity, it is going to the blood, everything is there. So we prepared a PAPI. PAPI means phytopharmaceutical API. So then we did in vitro in vivo on the PAPI, all in vitro in vivo, regulatory toxicology and the GLP guidelines we did, acute tox, repeat dose tox, general pharmacology, everything we completed. And then to go to the clinical trial, we need a formulation, robust formulation. And another thing is that since we are spending a lot of money in this, we need a patent on this. So getting a patent on the herbal medicine is the next another difficult task. So for that, we prepared a new novel formulation on this. So if you have seen the structure in previous slide, the Bhajan has a lactone moiety, so it is not stable in the intestinal pH. So to make the stable, increase the stability of the Bhajan, and that is the reason Bhajan has very low oral viability. So to improve that, we prepared a gastro-retentive sustained-release formulation. So these new capsules, they are getting floated in the GIT itself, and from here only there is absorption. And because of the swelling effect, buoyancy effect, it does not cross the pyloric sphincter. And based upon this we also validated in the PK studies. We did the comparative PK of the plain extract versus the sustained-release formulation and see the AUC, 3-fold increase in the AUC of the Bhajan in the plasma circulation. And we got IP on this, and this product we recently out-licensed to industry for launching in the market initially for as a dietary supplement for the management of pain in rheumatoid arthritis. And this we are developing in the phytopharmaceutical guideline. We already filed IND application to the DCGI to get the permission for doing the phase 1 clinical trial in healthy volunteers. So another example is the saffron. So if you know the Alzheimer's disease, this is the basic difference between the normal brain versus Alzheimer's brain. So this is the healthy neurons, and in the Alzheimer's brain, what happens, there are the two important pathophysiologies. There is a deposition of amyloid beta plaques in the synapses. There is a A-beta protein which gets aggregated, and there is a deposition. So it does not clear out of the brain, and that is the reason there is a loss of neurotransmission. And another important protein in this, the tau protein gets hyperphosphorylated and gets deposited as a neurofibrillary tangles inside the neurons. And because of both these approaches, both these hypotheses, there is a damage in the neurons, so many things happens, and there is a loss of memory. So there is a hypothesis published in 2010 when we started working on that. So there is a protein, efflux pump protein. So in the blood and the brain, there is a membrane is there. So there are the proteins which are actually regulating the export of efflux of this A-beta protein out of the brain to the blood circulation. So in Alzheimer's disease, both these proteins are down-regulated. So we thought that why not to induce this expression of this protein by small molecules. And while working on that approach, we did a lot of medicinal chemistry, and from these efforts, we identified that this natural product, crocin, is showing the increasing the A-beta efflux out of the brain. And based upon this, we started working on the saffron. So saffron contains this class of compound which are 16 crocins, and all crocins, when goes to the stomach, everything gets metabolized into the single molecule called as crocetine. This acts as a prodrug. So there are sugar moieties on both the ends, so they get cleaved in the GIT because of the esterase, and crocetine actually goes to the blood circulation and finally to the brain. So this is the structure. So this is the HPLC profile of the saffron, where the crocin one is the major compound. This pure compound also we tested in the transgenic model of the Alzheimer's disease. So this model, and here at the 10 milligram per kg, we fed the animals for 1 month, and this has reduced the A-beta burden, so many things you can see here. Then later on we develop a PAPI, like saffron extract we prepared. We did all in vitro, in vivo studies, and not only this, so we know that crocetine is the bioactive compound, so crocetine also we synthesize in good quantity, and this pure compound also we tested in vitro, in vivo, and we observed that this pure compound also shows the same level of activity like the PAPI. And here we build the confidence that this is the compound which is actually going to the brain also. So we did the PK also of this. So pure compound, when we take the saffron or even the crocin, see this PK. When we take the saffron, this is the crocetine levels. Crocetine is only actually going to the plasma, and also we check the brain levels also. In the brain also compound is going. Then after this making a new formulation, we got IP on this. We completed all GLP toxicology studies, and this product also, capsule formulation, we licensed to the industry for launching in the market as a dietary supplement, and we are going to file IND soon, very soon now. So I will conclude my talk here. So based upon these two examples, I think there is a huge opportunity for translating this scientifically validating the traditional knowledge, so that you can go to the phytopharmaceutical, as well as you can identify new natural products where we can pursue medicinal chemistry and discover small molecule leads to go to the this direction of the allopathic medicines. So I must acknowledge my students during last 10 to 12 years, and these are my biology partners, some of the outsourcing partners for GLP toxicology studies. Thank you very much.
I now invite queries, if any, from the audiences.
Yeah.
Hello. Thank you, Doctor, for giving us such an informative lecture. I have some questions on regarding the phytopharmaceuticals, which very well you have explained the things. Here you have informed that 4 biomarkers are required for submitting it to the DCGI or SITSCO. So I have a question, like when we have a formulation where 4 or 5 herbs has been used in a formulation, so in that case, how many molecules we have to target? Whether the multiples of 4 or the total 4?
So, since you are talking about the polyherbal formulation, so although this US Botanical allows the polyherbal formulation, but SIDSCO guideline, they allow only single plant. That is, I think, one of the bottlenecks. If someone wants to develop the polyherbal, that is not allowed, still not allowed under the SIDSCO.
Okay. So we have to target on the pure extracts.
No pure extract, but single plant,
Single plant
enriched
extract.
fraction of the single plant.
Okay. Okay. Thank you, Doctor.
Dr. Barate, thank you very much for a fantastic talk. It was fast-paced, but the data was fantastic too. So I really appreciate your efforts and sharing what you have learned and how we can go ahead. I have one query. In the later part of your slide, you were showing the PK data of the plant extract. And sometimes what we have observed in our own experience, that the stability of these plant extract or the phytocompounds in the plasma or in the blood is very, very less. It becomes literally non-detectable after 1 hour of injection. So how did you really get such a stable response even after 24 hours of the dosing?
So that is the reason I'm speaking about, like what kind of secondary metabolites are there. That is very important. If I think you are speaking about some maybe terpene glycosides or those kind of compounds, they are bound to get degraded, even the extract also. They get transformed to other metabolites or something like that. So it is very difficult to, those kind of metabolites we should not select under the phytopharmaceuticals, because ultimately you will face problem in the stability studies also. So
Right,
compounds should have very good stability, because then only you can correlate with the biological activity.
Right, the compound that you showed that is coming from Kesar is like very long flat compound and has a some core in the center, which probably would be get chewed by the compounds or the other proteases in the plasma. So I was thinking that the stability of that compound might also be low.
Exactly.
But
so
what you're showing is like a very strong response, which looks very fascinating.
So that's what I've shown. I think I'll just quickly go to I'll explain that. So this compound, whatever top one structure that which is actually present in the plant, this crocin, this is not stable. Even in the extract, after you check after 1 month extract, HPLC profiling, you lose the content of the crocin. So what I have shown in this PK studies, it gets hydrolyzed, and this whatever the crocin is there, a glycan part, that is actually we are detecting in the plasma, whereas the parent compound is not there. So it is a kind of a prodrug. So this parent compound is getting hydrolyzed in the GIT itself. It is not going to the plasma also.
So you are actually modifying the main compound.
We are not modifying, body is modifying.
Body is modifying. Yeah. But you're giving the prodrug to the animals.
Yeah. So and whatever that hydrolyzed product is there, that is actually biologically active. We have proved that by doing the efficacy studies on the pure compound, this compound. This is not present in the plant, but it is effective because it is getting converted in the body.
Thank you, Dr. Bharat. Thank you very much.
So if we do not have further questions, I would like to thank you, sir. And now Professor Anurag Srivastavji onto the dais to give his talk upon use of medicinal plants in cancer treatment, a learning from Ayurveda. Let's all give him a big round of applause. Professor Srivastav is currently the Chief Clinical Research Advisor at Patanjali Research Institute. Yes, sir.
Thank you, ma'am. Om. Param Pujya Acharya Ji, Param Pujya Swami Ji, learned, distinguished researchers from India and abroad. Let us talk about how the herbal medicine or plant-based medicine can help patients with cancer. I'll talk very briefly because it's a very wide subject. So for those who are not tuned to the biology of cancer and oncology, I would introduce some basic concept. So you all have seen a plant with some overgrowth. So pointer hai kya pointer? Can I have a pointer? Can you see? So this area in the center, is an overgrowth. Thank you. It is an overgrowth of some tissue. Similar and overgrowth here. So in biology, we call it cancer. This can be benign, when we call it benign tumor, where it remains localized to the area of origin, the organ of origin. It may be arising from the fat, then we call it lipoma, from fibrous tissue, fibroma, bone, osteoma, in breast, fibroadenoma, and so on. So one is benign, and sometimes it has the property to invade to other tissues. So you must have seen a plant, say usually people plant, Ficus religiosa, growing onto other trees. And you can see the roots going into the tissues. So this is called infiltration in biology. So when tumor arises from one organ and infiltrates like the roots of the peepal tree into the other part, then it is called infiltration. And this is the hallmark of cancer or malignant tumor. So the malignant tumor is characterized by infiltration like the roots of the tree from one part to the other. So this is the difference between benign and malignant tumor. So this is just for understanding for those who are not tuned to biology of cancer. And because of this property, just like this tree, which sucks the nutrition, the water, the nutrition from the other tree on which it is growing, like a parasite, then it will deprive the plant from which it is growing, right? So similarly, a malignant tumor will suck the nutrition from the organ of origin. Suppose it is arising in the mouth, so it will suck the whatever protein, fat, carbohydrate you are eating, and will deprive the organ of origin, and ultimately through these roots it can also spread to other organs. So this is the difference between benign and malignant tumor. It infiltrates into other tissues and then deprives the organ of origin by this infiltration. So in biology, we have two types of cell division. One is called mitosis, normal cell division, where one cell in a very organized, disciplined manner, just as you are very disciplined students of Patanjali Medical Institute or university, you follow some rules and regulations of the university or the institution you work. Here, these cells, like very well-behaved, disciplined students, they divide from 1 to 2, 2 to 4, and like that. And after 8 to 10 divisions, they have their own death. So they die their own death. This is called programmed cell mitosis or apoptosis, programmed cell death. So this is very similar to the computer programs which you download from the internet. Suppose you are searching some information on the Google or internet, and suddenly an ad comes. Download this program for free. New version of, say, Microsoft. You will download that, but on 29th day or 30th day, a message will come. This is the last day. If you want to continue using this program, you have to pay $30, right? So Mr. Bill Gates, or the originator of the program, has programmed it for dying on 30th day, right? So similar to that, this biology, the God or the nature has programmed these cells to die after a fixed number of divisions. And after that, they will die their own death. This is called programmed cell death. And this is important because if the cells continue to grow in our body, we will have overgrowth of every organ. So nature or God has made this very unique system of programmed cell death, also called apoptosis. And contrary to this, when we come to a malignant cell growth, here there is no discipline. So they are like, you can imagine like a mob or terrorist who are not following any rule, and they cause disturbance in the society or disturbance in the community. And from one cell, it may become 10, 10 to 30th, 30 to 100, and so on. And they lack programmed cell death. So they continue to grow. And you must have heard of a story of a Rakshas or Asur, Rakta Beach. So Ma Durga used to kill, and every time the blood of that Raktabij will fall on the floor, suddenly a new Rakshas will emerge. So similar to that, so it was immortal at Rakshas. Similar to that, cancer cells are like immortal Rakshas, where they continue to grow without any death of the cells. So that's a basic difference between normal cells, which follow some disciplined rules of the game, rules of the body, and indisciplined growth of cancer. So the cancer can be summarized or defined as uncontrolled growth, which is purposeless. It does not follow any purpose. Normal cell division has some purpose. Blood cells divide to produce red blood cells, white blood cells, platelets. When you have a cut or a wound on the skin or a bone, then those cells grow to heal that wound. And after the wound has healed, then they will stop, because the purpose of healing is over. Here, there is no purpose, and they continue to grow. They are indisciplined, and they will ultimately rob or deprive the cell of body of the origin and ultimately kill the host. Now, I'll just take you through some of the important cancers, and the most common cancer of human race is now breast cancer. It's the commonest cancer of human race. स्तन कैंसर मानव जाति का सबसे अधिक होने वाला कैंसर है, और ये तेजी से बढ़ रहा है. पूरे विश्व में इसकी संख्या बहुत तेजी से बढ़ रही है, और हमें इस क्षेत्र में बहुत शीघ्र और एक ठोस कदम उठाने होंगे. और इसीलिए हम एलोपैथी या मॉडर्न मेडिसिन की साइंस को इंटीग्रेट करना चाहते हैं. योग, प्राणायाम, आयुर्वेदिक, यूनानी और सभी एंशिएंट सिस्टम ऑफ मेडिसिन से, जिससे कि हम इस महामारी जो से स्तन कैंसर या ब्रेस्ट कैंसर कह रहे हैं, उससे कुछ हम जूझ पाएं और कंट्रोल कर पाएं. ये डेटा है पूरे विश्व का. ग्लोबोकैन, WHO has a branch which deals with the data about cancer. It's called Globocan, Global Cancer Data. So Globocan data of year 2020 has reported that in different countries, we have different incidence of cancer. If you see the top line, that is Brazil. So in Brazil, among males, पुरुषों में 1 लाख पुरुषों में 631 पुरुष में कैंसर हो जाता है. Out of 100,000 males, 631 gentlemen will get cancer in the country Brazil. Next comes France, 504. And then we have different states of United States of America. And please see the last 4 lines, the line in blue. They are from our own India northeastern states. The northeastern states, Arunachal, Assam, Manipur, सिक्किम, इनमें इतना अधिक कैंसर है कि इसको भारत का कैंसर किंगडम कहते हैं. The northeastern states of India are called the cancer kingdom of India because the incidence of cancer is highest in these states. And why? We'll have to think about them. Similarly, among ladies, if you count 100,000 ladies, out of them, in again Brazil, 474 ladies out of 100,000 will get cancer. ब्राजील में 1 लाख महिलाओं में 1 वर्ष में 474 महिलाओं को कैंसर हो जाता है. तो ब्राजील में पुरुषों में भी और महिलाओं में भी सबसे अधिक कैंसर की बहुतायत है. और इसके बाद फिर न्यूजीलैंड और फिर यूनाइटेड स्टेट्स के दूसरे स्टेट्स हैं. और फिर अगेन प्लीज नोट द लास्ट 4 ब्लू लाइंस, अगेन दीज आर आवर ओन नॉर्थ ईस्टर्न स्टेट्स ऑफ अरुणाचल एंड नॉर्थ ईस्ट. सो व्हाई Brazil and some United States has the highest incidence of cancer? They are predominantly meat-eating countries. मांसाहार यहाँ का मुख्य आहार है, और वो भी रेड मीट सबसे अधिक खाते हैं. गाय का, बीफ, पोर्क यानी पिग का और आदि दूसरे एनिमल्स का जो रेड मीट है वो बहुत आयत से खाए. उनका एक मेन स्टेपल डाइट ही, जैसे हम कहते हैं कि हमारे भारत में ज़्यादातर लोग या एशियन लोग रोटी दाल चावल खाते हैं, उनका मुख्य आहार ये है. तो वैसे ही उनका मुख्य आहार रेड मीट है, लाल मांस का भक्षण करते हैं वो लोग, और इसलिए उनमें कैंसर की सबसे अधिक बहुतायत पूरे विश्व में है. और वही स्थिति हमारे नॉर्थ ईस्ट के इन 8 प्रदेशों में भी है. वो भी लाल मांस का ही भक्षण करते हैं. उनके यहाँ पोर्क, बीफ आदि बहुत अधिक, मांस को भून कर बहुत अधिक खाते हैं. तो रोस्टेड रेड मीट इज द मेन कल्प्रिट cause of cancer, and this is shown remarkably by the data both for gentlemen as well as for ladies. So we have to think in those lines. If we want to prevent cancer, we have to prevent meat eating and go to plant-based diet. So some of the common cancers, cancer is a multifactorial disease. It's not caused by one single factor. Age is considered a very important factor. Why? Because cancer is predominantly a disease of the age, except for some blood cancers like leukemias in small children. Param Pujya Swami ji brought one small child, who had leukemia, but that is rare. Children do not usually have cancer, but it's mostly a disease of 40 plus or 60 plus age group. Old age का बहुतायत से कैंसर होता है 40 के बाद अधिकांश पुरुषों में और महिलाओं में. हार्मोन्स कई कॉज कर सकते हैं कैंसर, जैसे कि स्तन कैंसर, ओवेरियन कैंसर, यूट्राइन कैंसर आदि हार्मोन से रिलेटेड हो सकते हैं. फिर टोबैको, तंबाकू एक मुख्य कारण कैंसर का है और पूरे विश्व में करीब 40% कैंसर तंबाकू से होते हैं. About 40 to 60% cancers in the world are caused by tobacco, with or without some infections. 4 infections are important. They are all starting from letter H. So you can remember 4H cause cancer. They are the HPV virus, human papilloma virus, जिसकी वैक्सीन भी बन चुकी है और जिससे सर्वाइकल कैंसर का प्रिवेंशन में एक महत्वपूर्ण योगदान है. तो अगर 9 to 14 एज की स्कूल गर्ल्स को HPV वैक्सीन की 2 डोज दी जाएं, तो हम उनको HPV रिलेटेड सर्वाइकल कैंसर, ओरोफेरिंक्स कैंसर और जेनिटिलिया गुप्तांगों के कैंसर से पूर्णतया बचाव कर सकते हैं. तो HPV is the main cause of cancers of the cervix in girls, of the oropharynx and private parts in both gentlemen. Then HIV AIDS का वायरस, उससे एक स्किन कैंसर होता है जिसे कपोसी कार्सिनोमा कहते हैं. हेपेटाइटिस बी से लिवर कैंसर का संबंध है. और हेलिकोबैक्टर पाइलोराई, रिसेंटली ये रिसर्च ने दिखाया है, शोध ने दिखाया है कि हेलिकोबैक्टर इन्फेक्शन से स्टमक का कैंसर संबंधित है. So these 4 infections are related to carcinogenesis. So we can prevent by controlling these infections by vaccination or by treatment of these infections. Lifestyle is important cause and we'll talk more about it. Diet is again another cause for many cancers. Pollution, air pollution, water pollution, food pollution, even light pollution. So you have heard of so many pollutions, but we do not talk of light pollution. Light pollution और हिंदी में जिसके प्रकाश प्रदूषण कह सकते हैं, वो उसका अर्थ है कि हम सभी पूरी विश्व में जितने जीव जंतु, चिड़िया, तितली, मधुमक्खी सभी बने थे, भगवान ने बनाया था उनको 12 घंटे के दिन और 12 घंटे की रात के दिनचर्या के लिए. इसीलिए सूर्योदय होता है, 12 घंटे दिन रहता है और 12 घंटे रात्रि होती है. When Thomas Alva Edison invented the bulb in 1879, he disturbed this natural phenomena of living with 12 hours day and 12 hours night cycle. Now you will see that till late 11 p.m. people have lights in their house or on the roadside, in the offices, and they are doing either WhatsApp or FaceTime. So this has disturbed the natural living of 12 hours day and 12 hours life cycle. This is called light pollution. Please do not take it lightly. It has been shown that ladies working in the night shift have 3 times greater risk of getting breast cancer than those like you who sleep in the night. रात्रि बनाई गई थी सोने के लिए, दिन बनाया गया था काम करने के लिए. इसको हम लोगों ने प्रकृति में बल्ब बना के उल्टा कर दिया है और उससे बहुत सारी बीमारियां हो रही हैं. जो नाइट शिफ्ट में काम करते हैं या देर रात तक जगते हैं, उनको इंसोम्निया हो रहा है, रेस्टलेसनेस हो रहा है. अगर परिवार शादी हो चुकी है तो उनको डाइवर्स ज़्यादा हो रहा है, उनको स्टेरिलिटी, इनफर्टिलिटी हो रही है, बच्चे नहीं हो रहे हैं और उनको कैंसर की भी रिस्क बहुत अधिक बढ़ रहा है. क्योंकि ब्रेन में एक बायोलॉजिकल क्लॉक होती है, पीनियल बॉडी. वी हैव अ बायोलॉजिकल क्लॉक इन आवर ब्रेन regulated by pineal body. Pineal gland regulates many hormones which are then controlling hypothalamus and pituitary. So all the hormones are controlled by this biological clock system. If you are awake till late in the night, you are disturbing your biological clock which God has created in our brain. And that's why we are getting so many diseases, and this is called light pollution. So we should be careful about this. And then there are many genetic factors as well. So in English, the old English adage saying is, what when eateth, so he becometh. Jaisa khaye ann, vaisa rahe man. And the modern scientific research has confirmed that this is exactly true. So I'll take you to the ancient literature of Vedic literature. Yukta har viharasya yukta cheshtasya karmashu yukta sapnava bodhasya yogo bhavati dukha. अर्थात आहार आपका युक्त होना चाहिए. आहार means your eating, your diet. विहार is lifestyle. विहार दैनिक दिनचर्या आपकी. और चेष्टा और कर्म. चेष्टा means effort or endeavor. If your diet is correct, if your lifestyle is good, but if your efforts and endeavors are wrong, you are thinking how to evade tax this year, or how to disturb or cheat your neighbor or your friend, then your cheshta, your effort, your endeavors are wrong. And your karma or your actions should also be correct. So if your eating, if your lifestyle, if your efforts, endeavor, and your deeds and work, they are all appropriate, modest to the amount, then only yoga will take away all these sorrows and illnesses. So this is the essence. Just by doing yogasana and pranayam, please do not think that you will remain healthy. You have to keep your diet under control. You have to take sattvic diet. You have to have your lifestyle under control. Again, sattvic lifestyle, sattvic gyan, sattvic karma, and your cheshta and effort, endeavor should also be sattvic. Then and then only yoga will protect from cancer and all the illnesses, in fact. Diet is regulated and controls or is responsible for about 30 to 40% of all cancers in the world. And most importantly, taking fatty diet or obesity causing diet, alcohol, and diet which is low in fresh fruits and vegetables, especially colored fruits and vegetables, diet which is rich in red meat, lal mas. तो लाल मांस और फल और सब्जियों का कम सेवन करना और ऐसे आहार लेना जिससे मोटापा अधिक हो जाए और अल्कोहल यानी शराब का लेना, मदिरा पान करना, इनसे करीब 30 से 40% कैंसर हो सकते हैं. और रेड मीट से ब्रेस्ट कैंसर, बड़ी आंत का कैंसर, कोलन कैंसर, पैंक्रियाज, अग्नाशय का कैंसर, ये सब अधिक संभावना होती है अगर आप रेड मीट अधिक खाते हैं. तो रेड मीट इज रिस्पॉन्सिबल फॉर 4 different types of cancer in our body. WHO and World Bank has shown that obesity increases the risk of 13 types of cancer. 13 types of cancers are increased in those who are obese. And in America and Europe, some parts of Europe, obesity is like an endemic. It's pandemic of corona came and went. But here, obesity is almost pandemic, and it's occurring about 59 to 60% of the American population are obese because they are all eating McDonald's, burger and pizza, and which is rich in saturated fat and high rich in calorie and meat. So please take care of your weight. Otherwise, you are at risk of 13 different types of cancers. This was known to us, from the centuries in the Vedic literature. But in the modern science, a very important research published in Journal of American College of Cardiology in 2020 has shown that there are 2 types of foods. So they have classified the food according to modern science of 2 types. One which causes inflammation. शरीर में जैसे अग्नि लगा देते हो, इन्फ्लेमेशन कर देते हैं, वो एक तरह के आहार हैं. और अभी आपने कोरोना में सुना होगा कि इन्फ्लेमेटरी साइटोकाइंस का एक स्टॉर्म आ जाता है, IL-6, IL-17, TNF अल्फा आदि जो साइटोकाइन होते हैं जिनसे शरीर में इन्फ्लेमेशन, जलन जैसी होती. तो उनमें क्या आहार आते हैं? रेड मीट, प्रोसेस मीट, डिफरेंट टाइप्स का मीट, रिफाइंड कार्बोहाइड्रेट. रिफाइंड कार्बोहाइड्रेट का अर्थ हुआ मैदा, रिफाइंड, जिसमें कि जिससे पिज्जा, नूडल्स, बर्गर्स आदि बनते हैं. और याद रखें 3 तरह के, स्वामी जी भी बताते हैं, आचार्य जी भी बताते हैं, 3 तरह के सफेद जहर क्या हैं? मैदा, सफेद नमक और सफेद चीनी. ये 3 सफेद जहर हैं, इनसे बच के रहें. तो 3 white poisons in modern day food are the powdered or refined carbohydrate derived from wheat flour called maida, which has no fibers contents, then white sugar and white salt, common salt or white salt, NaCl. What are the foods which suppress this inflammation? जो इस inflammation को खत्म कर देते हैं, इस जो जलती हुई अग्नि को, इन्फ्लेमेटरी अग्नि है जो शरीर में, उसको भस्म कर देते हैं, वो है एंटी इन्फ्लेमेटरी डाइट. और उनमें देखा जाता है ग्रीन लीव्स, येलो फ्रूट्स एंड वेजिटेबल्स, फ्रेश फ्रूट्स, मोटे अनाज. पहले मोटे अनाज जैसे बाजरा, जौ, चना, ज्वार आदि सिर्फ गरीब मजदूर लोग खाते थे. अब वो कहा जा रहा है, अभी यूनिसेफ ने आपने सुना होगा, यूनाइटेड नेशंस ने डिक्लेयर करा है ईयर 2023 इज द ईयर ऑफ द मिलेट्स. भारत के राष्ट्रपति जी और प्रधानमंत्री जी ने भी इसको एम्फसाइज करा है कि इस वर्ष द ईयर 2023 इज कॉल्ड द ईयर ऑफ द मिलेट, मोटे अनाज का खाना. तो दीज मिलेट्स और the mote anaz are rich source of many fibers, many antioxidants, many vitamins, and therefore they reduce the inflammation in our body. And this study from American College of Radiology has shown that if you are meat eater, if you are eating these red diets, then your risk of heart disease and also cancer increases. If you take predominantly these green and foods which reduce your inflammation, your risk of cancer and heart disease goes down. So the very important data confirmed by hardcore science from America. So again, our Shastras teach us that there are 3 types of food: Sattvic, Rajasi and Tamasi. Ayu, Sattva, Bala, Rogya, Sukha, Preeti, Vivardhana, Rasya, Snigdha, Stira, Irdya, Ahara, Sattvic, Priya. So Sattvic diets increase your Ayu. Ayu means age, longevity. आपको दीर्घायु शतायु बनाते हैं. सतोगुण बढ़ाते हैं, the good virtues, आरोग्य, they give you freedom from disease. Sukh means happiness, Preeti, love and affection, Vardhana, they enhance. So sattvic food enhance your life, enhance your age, enhance your love and affection amongst each other, and they make you free from illnesses. They give you freedom from illness, आरोग्य. वो क्या है? रसीले फूड, रसीले जैसे जितने भी फ्रूट्स आप सोच सकते, दूध, रस, मलाई, मक्खन आदि. Yes, in some amounts we have to eat even butter or ghee, because all the hormones in our body are synthesized from the basic molecule cholesterol. So it, for about 20-30 years, the scientists from the West told us wrongly that we should not eat any cholesterol. Cholesterol is bad. That was wrong teaching. Now they are coming back to the truth that some cholesterol containing food is essential, because all the hormones in our body are synthesized from the cholesterol. अगर माखन खाने में कोई बुराई होती तो भगवान बाल कृष्ण माखन चोर न कहलाते. कोई भगवान का रूप कोई जहर को तो नहीं करेगा ना, तो माखन चोरी करके थे, माखन चोर खाते थे भगवान कृष्ण गोविंद, तो वो अच्छी चीज होगी, तभी उन्होंने उसका प्रचार प्रसार किया. दूसरे जो आहार हैं उनका राजसी कहते हैं उनको. कटु, अम्ल, लवण, अति उष्ण, तीक्ष्ण, रूक्ष, विदाहिना, आहारा राजश्रेष्ठा, दुख शोकामय प्रदा. तो राजसी खाने से हमें दुख होता है, शोक होता है, और आमय यानी बीमारी होती है. वो क्या फल हैं? कटु, बहुत कड़वे, अम्ल यानी खट्टे, बहुत ज्यादा खट्टे, लवण मीन्स नमक. So WHO research has now shown that we should not take more than 5 grams of table salt. One spoon, छोटा जो चम्मच होता है चाय का चम्मच, वो 5 ग्राम का होता है. इससे ज्यादा अगर आप नमक खाएंगे तो आपको हार्ट डिजीज, ब्लड प्रेशर, और अब रिसर्च शो कर रही है कैंसर भी होने की संभावना है. अगर आप लेह लद्दाख में जाएं तो वहां देखेंगे मुंह का और गले का और पेट का कैंसर बहुत अधिक से पाया जाता है, और वो लोग इतना अधिक नमक खाते हैं, चाय में भी नमक डालते हैं वो लोग. तो salted tea is causing cancer in Leh Ladakh part of Kashmir, and excessive salt intake is related to occurrence of many cancers. And this was told to us by Lord Krishna 5,000 years ago in Bhagavad Gita. We ignored this knowledge, and that's why we are suffering. We have the sorrow, and amay means illness. And then tamsi food. Yat yamam gat rasam puti parushitam cha yat uchchishtam chamedhyam bhojanam tamsa priyam. तो वैदिक संस्कृति में एक दिन को 8 पहर में बांटा गया है. तो एक पहर, 2 पहर सब जानते हैं, 2 पहर माने दूसरा पहर. तो अगर 24 घंटे को 8 से भाग दीजिए तो 3 घंटा आता है. 8 पहर हैं. तो एक पहर, ये इस सिद्धांत के अनुसार जिस पहर में आप भोजन बनाइए उसी पहर में आप खा लीजिए. अर्थात 3 घंटे के अंदर पकाया हुआ भोजन खा लेना है. अगर 3 घंटे के बाद खाया गया तो उसको भगवान कृष्ण की इस डेफिनेशन के अनुसार तामसी कहा गया है. तो जब से मैंने करीब 20 वर्ष पहले जब को सीखा इस श्लोक को, तब से मैंने डबल रोटी यानी ब्रेड का खाना बंद कर दिया है. तो सैंडविच या ब्रेड आप जो खाते हैं बड़े शौक से, तो वो समझ लीजिए तामसी है, क्योंकि वो आज बनाई गई बेकरी में, कल आपके शॉप में आई, परसों आपने खरीदी और 3-4 दिन तक फ्रिज में तो ब्रेड रखे ही रहते हैं आप. तो वो इस डेफिनेशन के अनुसार तामसी है, उसको न खाएं. So lifestyle and diet can control lots of cancers. So we have to practice yogasana, pranayam, do regular manual labor to keep our body weight under control, eat fresh fruits and vegetables, and cooking which does not cause any deep frying and roasting. Again, carcinogens are produced if you do deep frying. चाहे वो पूड़ी, पराठा, समोसा, किसी भी चीज को आप डीप फ्राई कर रहे हैं, वो ब्राउन हो जाता है, तो ब्राउनिंग मीन्स कैंसर कॉजिंग पॉइजन्स आर प्रोड्यूस्ड. एंड प्रेशर कुकर से स्टडीज हुई हैं कि अधिकांश विटामिन और एंटीऑक्सीडेंट्स जो हेल्दी न्यूट्रिएंट्स होते हैं वो नष्ट हो जाते हैं, इसलिए प्रेशर कुकर नहीं यूज करना है. प्लांट्स के बारे में थोड़ा सा वर्णन करते हैं. प्लांट्स को हम लोग 2 तरह से यूज कर सकते हैं. प्लांट्स फॉर थेरेपी ऑफ कैंसर, एस्टेब्लिश्ड कैंसर, एंड प्लांट्स फॉर प्रिवेंशन ऑफ कैंसर. So these are some of the plants. Vinca rosea और पेरीविंकल हिंदी में सदाबहार कहते हैं, और इससे कई एल्कलॉइड्स बनाए जाते हैं जैसे विंक्रिस्टीन, विनब्लास्टिन, विंडेसिन आदि. ये करीब 50 वर्षों से इस प्लांट के पिंक वाला जो विंका रोजिया है उसका उपयोग हो रहा है, और कई कैंसर में इसका बहुत अधिक लाभ पाया जाता है. कैनबिस इंडिका और भंगा जिसे कहते हैं संस्कृत में, भंगा से भी बहुत ज़्यादा रिसर्च हो रही है और इसमें कई एंटी कैंसर प्रॉपर्टीज पाए गए हैं. तो ये संक्षिप्त में हम अगर इस कार्टून में देखें तो मीठी नीम, करी पत्ता जिसे कहते हैं, जो सांभर में आप डालते हैं जितने साउथ इंडियन डिशेस में, ब्राह्मी, ये बहुत अधिक स्ट्रॉन्ग एंटी कैंसर है. सौंफ, फेनल, हल्दी, करकुमिन तो बहुत अधिक है ही, जीरा, पुदीना, तुलसी, तुलसी जी जिसकी आप पूजा भी करते हैं, धनिया, नींबू, नीम, लेमन ग्रास जैसे बोलते हैं, और अश्वगंधा, नीम, ये आदि सब कैंसर रोकने के बहुत महत्वपूर्ण हर्बल हमें गिफ्ट भगवान ने दी हैं, और इनका हमें अधिक मात्रा में उपयोग करना चाहिए जिससे कि हम कैंसर जैसी घातक बीमारी से बच सकें. नजेला सटाइवा, कलौंजी, कलौंजी का बहुत वर्णन है यूनानी में भी, आयुर्वेद में भी, और पैगंबर मोहम्मद साहब ने तो ये बताया कि कलौंजी से मौत के अलावा सारी बीमारी ठीक हो जाती हैं. सिर्फ मौत को छोड़ के बाकी सारी बीमारियां जड़ से ठीक हो जाती हैं अगर आप कलौंजी का नित्य सेवन करें. तो कलौंजी में बहुत मॉडर्न साइंस में भी रिसर्च हुई, कई लोगों ने पीएचडी करी, उसमें देखा गया इसमें एंटी कैंसर बहुत अधिक है, एंटी हार्ट डिजीज का बहुत अधिक महत्व है. तो कलौंजी में बहुत अधिक लाभवर्धक गुण हैं, इसको सेवन करना चाहिए. टैक्सेन, टैक्सेन आजकल हम लोग कैंसर में टैक्सेन ड्रग्स यूज करते हैं, पैक्लीटैक्सेल, डोजीटैक्सेल, वो एक प्लांट है टैक्सस, उसकी छाल से पैदा किया गया है. तो टैक्सेन बहुत, जिंजीबार यानी जिंजर यानी अदरक, थाइम, अजवाइन, टमाटो में लाइकोफिन्स होते हैं, लाल टमाटर में लाइकोफिन नामक कैंसर से बचाव के तत्व पाए गए हैं. गार्लिक यानी कि लहसुन, नीम तो हम जानते ही हैं बहुत अधिक औषधीय गुण हैं और उसमें कैंसर से भी लड़ने की क्षमता है. रेड ग्रेप्स में रिजर्वेटॉल नामक एक पदार्थ है और इसका ये बोटैनिकल नेम है. तो इन सब जड़ी बूटियों में, इन सब हर्ब्स में, फलों में बहुत अधिक कैंसर से लड़ने के तत्व हैं. ब्रोकली पे काफ़ी रिसर्च हुई है स्पेशली अमेरिका में, और इसमें देखा गया है कि ब्रोकली से ब्रेस्ट कैंसर और दूसरे कई कैंसर से हम बचे जा सकते हैं. लेकिन ब्रोकली को थोड़ा कुक करके खाना चाहिए, कच्चा नहीं खाना है, क्योंकि देखा गया उसमें जो कैंसर निरोधक जो गुण है वो फाइटोकेमिकल सल्फोराफेन है, और सल्फोराफेन सबसे अधिक बढ़ जाता है अगर उसको थोड़ा सा कुक करके खाएं। इससे कैंसर सेल की ग्रोथ रुक जाती है और रिसर्च में देखा गया है जो जीन है जिसके द्वारा कैंसर की सिग्नलिंग हो रही है, जो नॉर्मल सेल कैंसर में परिवर्तित हो रहा है, वो जीन्स भी रुक जाते हैं, थम जाते हैं और कैंसर फॉर्मेशन की क्षमता भी रुक जाती है। तो ब्रोकली खूब अधिक मात्रा में, जो हरी गोभी जिसे कहते हैं हिंदी में, ब्रोकली खूब अधिक मात्रा में सेवन करें। ये एंटी कैंसर हर्ब्स हैं, लाल पत्ते, हरे पत्ते, जो रंगीन सब्जियां हैं वो बहुत अधिक मात्रा में लें। लेट फूड बी दाई मेडिसिन एंड मेडिसिन बी दाई फूड। ये हिपोक्रेट्स जो मॉडर्न मेडिसिन के, जो एलोपैथिक मेडिसिन के जो गुरु कहे जाते हैं, फादर कहे जाते हैं, उन्होंने ये कहा है कि फूड को मेडिसिन के रूप में। और हमें अभी हमारे जो प्रिंसिपल साहब हैं डॉक्टर अनिल साहब, वो बता रहे थे सुबह कि आहार सबसे बड़ा सर्वोत्तम औषधि है। तो वो हमें समझना चाहिए कि आहार सर्वोत्तम औषधि है और खाने को आप आहार को एक दवाई के रूप में लें। कुक लाइक दादी माँ। जी हाँ, जो मॉडर्न कुकिंग है तो प्रेशर कुकर की कुकिंग और दूसरे यंत्रों से कुकिंग, जैसे जो सिजलर वगैरह जो बना रहे हैं, बारबेक्यू कर रहे हैं, वो सब बहुत हानिकारक हैं, उससे कैंसर पैदा होने के गुण बढ़ रहे हैं। तो दादी माँ जो कुक करती हैं वो देखिए कितनी प्रसन्नचित्त हैं, वो तो प्रसन्नचित्त होकर बनाया हुआ खाना बहुत लाभप्रद होगा। और देखिए बैठ के हाथ से आराम से बना रही हैं। चक्की मत इस्तेमाल करें, मिक्सी, चक्की इस्तेमाल करें, मिक्सी नहीं, क्योंकि उससे थोड़ा आपको मेहनत भी होगी, आपका वजन भी कंट्रोल होगा और जो दाने होंगे वो स्वास्थ्यवर्धक होंगे। और जहां भी आपको अवसर मिले एंटी कैंसर हर्ब्स और प्लांट्स लगाएं। ये हम लोगों का एक स्वास्थ्य का एक प्रयोजन था, तो हम लोगों ने जो एंटी कैंसर पौधे लगाए, जैसे तुलसी, कलौंजी, हल्दी, लेमन ग्रास, अदरक, गार्लिक, ब्रोकली, सदाबहार आदि। तो जहां भी आपको मौका मिले घर में लगाइए, बाहर बाग बगीचे में लगाइए। एंटी कैंसर हर्ब्स का खूब अधिक प्रयोग करिए। कैसे हम प्रिवेंट कर सकते हैं कैंसर से? वजन कंट्रोल करके, रेगुलर एक्सरसाइज, रंगीन फल और सब्जी खा के, से नो टू टोबैको एंड अल्कोहल। एंड for breast cancer we can also prevent by having breastfeeding the baby for at least 2 years. So I conclude my talk here. Thank you very much. And if there are any questions, please let me know.
Thank you very much, sir, for this excellent and enlightening talk. I had one question.
Yes,
what you mentioned that in the northeastern region, there is a lot of cancer that is happening from the food. And most of these people are eating a lot of roasted food. And what is related is that acrylamide and glycemide is accumulating a lot in their body. So would you consider that, number one, the question is that, number one, that not only for the non-vegetarian food, but the vegetarian foods which are mostly fried and they are grilled, they are actually highly toxic to the body. That's one thing. The second thing is there any research that has been done on the type of food that is being taken up by the people and that is related to the microbiota present inside the stomach, which might be related to the development of cancers?
Thank you, sir. So, अधिक फ्राई करने से, deep frying or roasting generates some chemicals called nitrosamines. So these nitrosamines are carcinogenic. Many animal studies have proven this fact. Then taking foods along with tobacco and alcohol and betel nut. So in the northeast, there is ample use of both tobacco and betel nut. If you go to city of Guwahati, Gua means Supadi, Hati means bazaar. So Guwahati is the largest, biggest bazaar of Supadi. So Supadi has been confirmed. The betel nut or areca nut has been proven to be a class 1 carcinogen. And if you use betel nut from your childhood in pan or in Supadi or as such, along with other food items, that is increasing the risk of cancer. Similarly, infection like Helicobacter pylori increase the risk of stomach cancer and GI cancer. So yes, you are very right. Some microbiome are related to occurrence of cancer, and Helicobacter is one of them. And hepatitis B, again, is going through the gut only, going to the liver, causing liver cancer. So some infections are related, and that's why there's a lot of research on microbiome related to various cancers. Thank you.
Thank you so much, sir. I would now like to invite our next speaker, Professor Partha Roy-ji, in the cure and management of cancer, a mechanistic approach. Presently, Dr. Roy is a professor in the Department of Biosciences and Bioengineering at Indian Institute of Technology, Roorkee, India. His current research interest is development and validation of nutraceuticals for the cure and management of various diseases, with special emphasis on diseases like cancer, diabetes, and bone-related disorders. Professor Roy has published more than 170 research papers in peer-reviewed journals and has authored several book chapters. Please give him a warm welcome.
Respected Swami ji, respected Acharya ji, Dr. Anurag, I have a deep gratitude for inviting me to this occasion. It's always a pleasure and honor to be here in this complex, because it's really giving me a great feeling. More than I speak, more I learn from here. So I take back more than what I give here. So I'm really honored and thankful to all of you for giving me this opportunity. So my topic is this role of some phytochemicals which are used in the Ayurvedic formulations for the cure and management of breast cancer and associated problem. So in this topic, I will try to cover up the story that cancer progression, breast cancer prevention, or associated problems, how it can be controlled. So these are, I think, I can easily bypass because my earlier speakers has already given very elaborative discussions about this. Our laboratory work on endocrine-related cancers, mainly breast, prostate cancers, ovarian cancer, cervical cancer, so some of these cancers. So today I'm talking about the breast cancer, so I will skip these slides to save some time. And this is a slide which shows how the breast cancer progresses. Again, I think my previous speaker, respected Srivastav sir, has talked hugely about the progression of the cancer and explained everything. So I can easily bypass. But what I can summarize here is there are two things I will talk about. I will talk about the cancer happening and the metastatic process. So coming to the existing drugs that is available for the cure of breast cancer are this. There are targeted therapy-based drugs, there are chemotherapy drugs, and if you see, there are lots of side effects. Everything has side effects. So this actually promotes us to look for the alternative approach for cancer therapy. Now, if you see the phytochemicals just I was talking about, so he just made a very good base for me, so I don't need to worry about anything else. So your food can be your medicine, not the medicine be your food. So with this approach, we work on some of the phytochemicals, which I'll talk about just now. So as the initial, when we started this project has been going in our lab for maybe almost like 10, 12 years. We have published, we have completed about 4 PhD students with this work, but I am giving only a glimpse of it. So when we started it out, we selected some of the plants, very important plants, and most of them had some literature information about the anti-cancer properties. And when we proceeded with this, we found that among them, most of them showed the anti-cancer properties, but Pterocarpus marsupium, shown over here, showed a very potent anti-cancer property. And then we went back to the literature and wanted to see what is this Pterocarpus marsupium was. And we realized it consists of a very important phytochemical that is pterostilbene, and it has a long history of usage in various Ayurvedic formulations, and it was sold commercially in US as pterostilbene for various health beneficial effects, which is summarized over here. So we started working on this pterostilbene, and when we saw the literature, we found it has a very close relationship with another molecule called resveratrol. So the basic difference between terostilbene and resveratrol is that it is dimethylated analog of resveratrol. Now, what is resveratrol is? It is basically a plant metabolized, and it has got known to have large number of health beneficial effects. And resveratrol has been used for various health beneficial activities as summarized here. If you see, it can cure all these effects. So, but it is still not very popular as a drug or as an phytochemical because of several reasons. Its bioavailability is very low, high dose required for showing the effect, toxicity is there, and complex mechanism of action. So this prompted us to see, can we use terostilbene as a drug or as a nutraceutical for our future use? So while we had been getting some good results out of that, we realized, as just now Sarah was telling, that in broccoli, cauliflower, especially in the broccoli, there is one residues which we found was that isothiazide residues, which also has known to have a very potential anti-cancer properties. So we made some studies using these particular residues in our studies, and we found that it shows potent anti-cancer properties. Then while we had been working on this, so we realized that this is isothiazide residues have very important properties in the phytochemical because it metabolites and hydrolyzed in different forms, and it has got a large list of biological activities. So that actually forced us to think in a different way. We moved to a reverse pharmacology approach, where we took that, okay, what is the mechanism of resveratrol, terostilbene, and what is the mechanism of isothiazide residues. So we thought that initially we had been purifying this compound from the plants, but we thought we can take a different approach, because if we can combine them together, what is the effect of that? So actually one synthetic chemist helped me in this process, and he designed a very nice molecule for us, where he made a hybrid molecule of two phytochemicals combined together. Although it became like a drug, but we wanted to see what would happen if they are present in a complex form within a system. So we got a new hybrid compound and we want to test it out in the systems, and we made an elaborative experimental thing to establish this particular molecule in our future experiments. Now, when you see the literature, if you see in Ayurvedic medicine, terostilbene and resveratrol both has been used for a long history of their usages there. So this gave us a boost and support, okay, we are moving a right approach, that we are using a molecule which is really important from the pharmaceutical point of view. In this process, we published a large number of publications using terostilbene as an anti-cancer agent, using various models, in vitro, in vivo models. But now we move toward the next phase of it. We wanted to see how this hybrid compound, when you combine two pharmaceuticals together, how they behaves. So our initial experiments are very simple. We started with MCF7 cells, which basically is an ER-positive breast cancer cell line, and we could show that this drug was basically causing showing the cellular proliferation was inhibited by this particular drug as compared to normal terostilbene. So it was more potent in all terms of cell proliferation, in terms of causing the cell death, and it was causing a severe cell deaths in the cells as compared to the normal conditions. So based on we performed several experiments to show this, that it was killing the cells very efficiently, and it could stop the cells in the G2 and M phase arrested condition. Typical characteristic feature of any anti-cancer drug, it could break the DNA ladders. And next question was that, if you're killing the cells, what is the mechanism for that? And it was basically in caspase dependent, but we wanted to see, because caspase happens in two different ways, in an intrinsic pathway as well as extrinsic pathways. And we tried to see which pathway is involved, either these or both of them. And we could find that it was involving only caspase 9 and 7. That means it was involving the intrinsic pathway, and extrinsic pathway is not there, because if you block the extrinsic pathway, then anti-cancer property was still there in the drug. So this shows that this particular compound was acting through an intrinsic pathway, not involving extrinsic pathway. And we could see that dose-dependently, this caspase 7 and 9 markers of, these are the markers of intrinsic pathway, they are getting enhanced. So this was continuing, and this work we continued, and we could show the different markers like BCL, BAX. This is the hallmark of the anti-cancer activities. They are all upregulated, and the cancerous, these cells were, like you can see here, survivin was downregulated, so that means the cell was dying out. We could show the ROS generation in the cell, so you can see that ROS generation in the cell was there, which killed the cells. And we could see the cytochrome C production was also very high in these cells. So these cells were really dying in response to these particular molecules. Then while designing the pathways, which pathways were really involved, we could see that AKT and ERK dependent pathways were involved, and they got down-regulated in response to these particular drugs. And it was in a drug, and it was both in a time-dependent way. We could see the phosphorylation of this AKT and ERK were down-regulated in these cells. So in response to these drugs. So this drug was really miracle one. And when we blocked these pathways, we could see the additive effects, because in response to the inhibitors of AKT and MAP kinase, we could see that inhibition was further down-regulated. That means it could combinedly affect the cell proliferation process. So, and then next phase, we wanted to explore the other part of it, that what happens if the cells become resistant to drugs, because breast cancer therapy becomes difficult because the cells become resistant to these drugs. And we could show that in the normal condition, the cells become resistant, they form the spheroids. But once the cells are treated with this phytochemical composition, we could see the cell spheroid formation was reduced, colony formation was reduced, and the cell was responding to the drug. So we could revert the drug-resistant conditions also from the cell. So in the next phase, we tested this in the animal studies. We developed the tumors in the animals and treated them with the phytochemicals, and we could see that in response to these particular chemicals, by the 30th day, the tumor was almost gone, and it was, while in the case of non-tumor control, it was quite high, so it could reduce the tumor formation in the animals. So based on this, we could show that it was involving the AKT-dependent pathway and ERK-dependent pathway, and it was inhibiting the caspase-dependent, it was stimulating caspase and inducing the tumor, inhibiting the tumor progression in the cell. So when this was going on, then my another student, this was our work of Nikhil, one of my PhD student, and then my another student joined in, and he wanted to see the effect on the MDMB231 cell, because we established it is anti-cancer properties there, but now we wanted to see what happens, the high aggressive breast cancer cell, MDMB231 cells were there, which is an aggressive breast cancer cell, and which is responsible for metastasis. So we could see that this particular drug was inhibiting the progression growth of these cells, and it was also inhibiting the migration of the cells. Both cell inhibition migrations were inhibited in response to this particular drug, and cellular morphology was also altered. So we could see that this drug was not only affecting the normal proliferation, but it was inhibiting the anti-cancer metastasis of the cells. So we could establish that it was acting on the MDMB, which is basically a marker for metastatic breast cancer cells. It was inhibiting them as well. So in the next phase, we wanted to see whether it was inducing apoptosis or not. We could see that this was inducing apoptosis in the cell, as shown by the AOEB staining and DAPI staining. You can see the formation of the apoptotic bodies in the nucleus in response to the drug, and that's which was absent in the normal cells. It was specific to these cancerous cells. So we could say that it was actually inducing the apoptotic death in in these metastatic cells as well. Now the question was, if it was really preventing the metastatic process, how it was doing it. So we know there are different markers, twist, Snell's, CYP1 are the different markers which induces the metastasis in the cells. So we wanted to see how if we can see this particular drug was inhibiting the epithelial to mesenchymal transitions or preventing the metastasis in the cells. So to see this, we targeted several pathways which are responsible for inducing metastasis. So we targeted NF-kappa B, which is a very critical player in any metastatic process. We targeted Snell, we targeted twist. So 3 different culprits responsible for metastasis in the cell. So when we see this, as you can see here, in response to these particular phytochemicals, we could see there was an upregulation, the localization of the zona occludin, response marker for the metastatic inhibition was there. And in addition to that, we could see that all these epithelial markers were upregulated, while the mesenchymal markers, metastatic markers were downregulated. So that means this particular drug was really acting on the cellular system to establish the cells towards the epithelial form from the mesenchymal form. So that means it was a pure indication of anti-metastatic properties. Finally, Snell and twist were also downregulated in response to the phytochemicals that you could see. It was significantly downregulated in response to this particular drug. So this was inhibiting the formation of metastatic process in the cells. Now the question was how it was doing it. So we wanted to next understand what is the exact mechanism of inhibition of metastasis. So we wanted to see what was the role of NF-kappa B in the cell, because one process could be, it could inhibit the production of NF-kappa B, or it could inhibit the migration of NF-kappa B from the cytoplasm to the nucleus. So we wanted to address this question in the next phase of our study. So what the first thing that came to our mind was that, what was happening to the NF-kappa B in the cells? So if you see this data, you can see the relative level of mRNA expression in the cell was not changed. That means this particular phytochemical was not altering the NF-kappa B expression. So if it is not inhibiting the expression, then what it was doing? Then we did a very important experiment. So we could see that it was restricting the NF-kappa B, because to do the function, NF-kappa B has to migrate to the nucleus. But this particular phytochemical was inhibiting the migration of NF-kappa B to the nucleus. It was retaining in the cytoplasm. So if it is in the cytoplasm, it is unable to act on the system. So naturally, we could see, and this was shown over here, as you can see in the control cell, it was in the nucleus. But as soon as you treat them with the pterostrelvin, this phytochemical, it was located in the cytoplasm. So it was migration in the nucleus was inhibited, and that's how it was inhibiting the function of this. And this was again showed over here. So in the next stage, we wanted to see how it was doing it. So this is the site promoter for the twist and the snell, where the NF-kappa B goes and binds and induces the expression. And we have already established that NF-kappa B migration was inhibited by this particular phytochemical. And this was shown by a chip assay. So chip assay shows that binding of the protein to the nucleus, to the DNA sites. And you can see here, in response to these particular phytochemicals, the binding of this new NF-kappa B to the promoter region was completely inhibited. So this showed that inhibition was there. So as a result of it, the expression of these twist and snells were inhibited. So we could say that treatment prevents the nuclear translocation of the NF-kappa B, and available NF-kappa B in the nuclei for binding to snell and twist promoters is reduced. As a result of it, the cells, the function, the action of twist and snell were reduced. So we could show that NF-kappa B was inhibited in the cytoplasm, so it was unable to migrate to the nucleus, so snell and twist level was low. Naturally, the EMT was reversed and apoptosis was induced. So, so this was the question was, now the question was how PTR-ITC was inhibiting the NF-kappa B from migration to the nucleus. So this was a very important thing we wanted to understand. Now we looked at the next step of it. So we did a systematic study to see what happens actually. This is a protein I-kappa B beta, and it has got a NEMO binding domain, and NEMO is basically I-kappa gamma. So this NEMO protein interacts with this one, this I-kappa beta, and this forms a complex. And once this complex is there, they actually phosphorylates I-kappa B alpha and sequestrates out. So once it sequestrates out, so this NF-kappa becomes free, and then it migrates to the nucleus. And once it migrates to the nucleus, it activates snell and twist. So this was actually the normal process that goes in a cell. So what we could find in our study was that, so this is the transcription of snell and twist, and that causes the metastasis of the cells. So what we could see in response to this pterostrelvin was a little bit different. What we could find was this. So it was inhibiting the binding of interaction of NEMO and I-kappa B beta. As a result of it, once this complex was not formed, so this I-kappa beta was never phosphorylated. It remains together with the NF-kappa B, and since it was bound, it was never free, so NF-kappa was not free to bind to the nucleus, go to the nucleus. So naturally, the transcription of snell and twist was downregulated. So this is our hypothesis, and we established this by a docking study, and where we could show very systematically that it was inhibiting the binding of this PTR-ITC was blocking the NEMO binding domain in the I-kappa B beta, and thus inhibiting the binding of NEMO to the I-kappa B beta. And that's how it was inhibiting this process. So this was a summary that we could see, and this was experimentally shown also. So when we see this, and you can see it here, so when you treat the cells with this particular drug, and we could find that interaction between the NEMO and I-kappa beta was reduced, and both by reverse chip, we could show that it was completely inhibited. So this actually established our hypothesis that inhibition of NEMO and I-kappa beta was done by this PTR-ITC, and that's how the cells could not do. And this is another proof to establish that we could show that phosphorylation of I-kappa beta was also inhibited. So that is why it was not getting sequestered. It was binding to NF-kappa B and inhibiting its migration to the nucleus. So this actually again prove our hypothesis that why it was unable to, NF-kappa B was not migrated to the nucleus, because I-kappa B was remaining bound to the NF-kappa B, and this was done by the PTR-ITC, and it was actually inhibited action. So this was a summary of what we could find, that NF-kappa B binding to the nucleus or migrating to the nucleus is inhibited by this particular drug, and epithelial mesenchymal transition was enhanced by this particular phytochemical complex. So to establish this, we performed a series of other experiments in our next studies. We developed tumor in the animals, breast tumors and other tumors in the animals, and we could see that in response to this particular drug, the tumor volume was greatly reduced in these animals. And what was interesting, and we could see that the markers of tumor markers, that is immunohistochemistry to show the tumor markers in the cells, are also completely inhibited in response to these particular drugs. And what was most interesting in this process was that in the breast cancer leads to the metastasis to the different tissues. And this was seen in these animals which were having tumors. They had the tumors in the lungs also, because breast cancer cells migrate to the to the lungs, and it was shown over here. But the animals treated with these particular phytochemicals, they had a very reduced level of tumors in the lungs. So they are all gone. And if you see the architecture, morphology of it, so this was the normal lungs of the animals, but in the tumorous condition it's like this. But when the animals are treated with these particular phytochemicals, and you can see here, it's almost close to the normal animals. So this shows that this is a very important, potent inhibitor of metastasis using the in vivo animal models. Next was that we wanted to see what is happening in the bones formation or bone metastatic process. And as we can see here, bone modeling is also controlled as a metastatic process, because breast cancer cells migrate to the bones to cause the metastasis. So what we did in these animals, we could see the cortical bones that were really normal conditions, but when the animals has a tumor, they had the cortical bone condition or destroyed. And when we treated the animals with our phytochemicals, you can see the bone remodeling was taking place. So these bones again came back. So this again showed that metastatic bones are again recovered by these particular phytochemicals. We had different mechanisms to show this. We established this, and we published this data also in our different studies. So in order to further establish this hypothesis, we created the dexamethasone-induced bone morphology models. And you can see when the animals are osteoporotic, this was like this. But in response to our phytochemical, the bone remodeling was taking place. So this is not only helping in preventing metastasis, but it was also inducing the bone formation process. So this was a very important finding. And we could see here, if you see the morphology of the osteoporotic conditions, this is like this. But in response to the phytochemical, it was also inducing the bone formation. So if there is any damage in the bones by metastasis, it could be recovered by this. And in the next phase, we wanted to see the strength of the bone. So we showed that because bone formation, recovering the bone is one, but does the bone gets back to its original form or not? So we tested the bone strength of these animals, from the animals, and you could see the bone strength was coming back, which was lost, it has coming back. And architecturally, bones also looked very close to the normal conditions. So this was not only recovering the bones, but it was actually bringing the bones back to its original conditions. So we saw the different markers of the bones. These are very common bone absorption markers that we see everywhere. It was all improved in these animals. So I'm not going details of this. We just recently published this paper in Biochemical Pharmacology, where we established these entire pathways. And also we performed the toxicity analysis for this, analysis, and we could see there was no apparent toxicity in the liver and kidney of these animals, which we made. So that was not toxic at all. So, and finally, we conclude that it specifically inhibits metastatic breast cancer cells without affecting normal cells. It reverts endothelial mesenchymal transitions, and it inhibits NMO and I-kappa binding interactions, resulting in prevention of NF-kappa B nuclear translocations, and it can inhibit the metastatic regulatory like external twist. So this is my research group people. Vinay is the person who did the last part of the work, who major part of it. And in addition to these other students in my lab, they are my current students, they are there. Vinay just finished and he has gone to the US now. He is now in Albert Einstein Medical School as a postdoc. Parul is another student who also finished just PhD. She's moving to Stanford just now. Sandeep is Augusta University now. So these are my current PhD students at the bottom of it who are doing in different projects. And this shows, so I presented a part of my work done by Dr. Nikhil and Dr. Ajanta. Both of them are my PhD students, earlier PhD students. and some part are also done by Somesh Banerjee. So these are my collaborators with whom I collaborate in various aspects, and of course, Dr. Bhajan is my friend, philosopher, and guide in different things. I learn Ayurveda and all these things from Anurag. So thank you, Anurag, for providing me, helping me out in this. Thank you very much. So thank you. That was all from my side. Any questions? Yeah.
Good afternoon, Professor Rai. I'm a clinician. I do not know anything about your molecular biology, but 20 years back I knew about this resveratrol.
Yes.
Though it is not available in the clinical practice, but the use of it is not in the grape of seedless grape.
Yes.
It is in the red grape skin
Yes.
of the red grape that we used to know.
Right.
And it has a very beneficial effect in diabetes.
Yes.
In diabetes, cardiovascular risk factors,
Yes.
and also it is said that the survival period is also increased. I think the principle of this particular drug was the anti-inflammatory,
Yes.
antioxidant,
Yes.
removing as a scavenger of the ROS,
Yes.
reactive oxygen species. Secondly, what you said about the isothiocyanate, sulfide, I think sulfur bond is there inside
Yes.
it, which is responsible. What I tell to my patients that do not take the cabbage,
Right.
cauliflower, all the crucifera family things, because it will increase your thyroid gland. The thing, how does it work? Now I come to know, and I knew that it causes the killing of the cells, the
Yes.
thyroglobulin, follicular cells, those things.
Yes.
If they are killed, then what will happen? The hypothyroidism will, TSH will be increased, and that will
Balance.
hyperplasia of the gland and these things, hypothyroid. So I used to tell my patients that not to take all those things.
Yes.
And now today you are telling, I didn't know all this molecular thing, and the benefit of this, combining these two things,
Yes.
resveratrol and the isothiocyanate,
Yes.
both of the things, it will be beneficial in the carcinoma like this thing, where it will having the same principle, I think. The anti-inflammatory, antioxidant, killing the cells, the same as it is the isothiocyanate in the thyroid, the same principle. I didn't know about your molecular thing. In a clinical practice, we used to say all those things to me.
Yeah.
Thank you very
Thank you,
much
sir.
for your educative.
Thank you. So actually, all clinicians are the base. We got the inputs and we tried to give it a scientific background to this one. That is what. Thank you so much, sir.
For the last 30 years, I am practicing medicine. So thousands and lakhs of patients I have seen, and I have prescribed whatever I told you.
Okay.
Thank you very much.
Thank you, sir. Thank you.
Dr. Roy, a fantastic presentation as usual. You put the science at the fastest way possible, and we are really lucky to have you back here. And every time, whatever you speak, we learned a lot. And your recent data on the bone restructuring, it was really marvelous. So thank you very much. And kudos to whole of your team who has worked very significantly. I had just one query, that when you're looking for this bone remodeling or restructuring, I mean, sometimes these also go out of the hand. That means sometimes they overgrow.
Yes.
And that also lead to a problem, right?
Yes.
And that lead to other issues, and you have structural artifacts
Yes.
in animals or in the human.
Yes.
Is there any way to check it by the data that you have that this would be a controlled process and it will not overshoot from the defined limits?
Yeah. So thank you, Dr. Anurag, for asking this question. It's a very important and pertinent question. So because, bone remodeling, what you have said, is a very complex process. A balance between osteoclast and osteoblast is really a critical problem. So if it overdo, it will have a very serious effect. That's what. So what we did, I did not show here. We have tried to balance it out with another way, osteoclast differentiation process. So there is a minor osteoclastic proliferation at the initial stage. If the osteoclast is there initially, then it will kill the overgrowing bone cells. So, but if you give the drug dose increases, then osteoclast goes down. So that means a dose is definitely required. That means if you overdo the drug, it will go to the bone, overgrow the bone. But it's minor dose, whatever dose we have tried, at this concentration, osteoclast goes and then it drops down. Osteoclast initiates the formation of bones and then the bone resorption follows out. So a cycle of osteoclast and osteoblast is always there. So that is controlled process. Thank you.
Thank you very much, sir. Dear students, your inquisitiveness and interest throughout the session is really commendable. I request all of you to please continue maintaining it and keep your movements limited. Now I invite the last speaker of this medicines that could combat cancer. Dr. Lochhabji is Senior Research Scientist, Drug Discovery and Development Division at Patanjali Research Institute, Haridwar. Om.
परम पूज्य स्वामी जी की सूक्ष्म उपस्थिति, परम श्रद्धेय आचार्य जी के श्री चरणों में मेरा नमन. We are delighted and honored to have the pioneers, experts of Ayurveda, medical sciences, ethnopharmacology, basic research, experts on the same platform today in the third series of Plants to Patients, and we would be more delighted if you would be able to join in the fourth series as well. So, a very good evening, everyone. I am Dr. Savita Lochab, and I would be giving you a brief outline of the cancer research that is going on at Patanjali Research Institute. So if I talk about the cancer statistics, then it's been a decade that every year cancer remains the leading cause of death. And according to the Globocon 2020, the number of new cancer cases diagnosed in 2020 was 19.3 million. It's a huge number. And most devastating is that it claimed around 10 million lives due to cancer. So if you look at this world map where this blue one, it shows the rate of incidence of cancer, and the orange one shows the mortality rates, and it clearly shows that how cancer is engulfing the world. These cancer statistics are not only numbers, but they are prepared with a thoughtfulness. They actually guide the researchers to develop the new preventive measures, to develop the new therapies, treatments, so that these statistics could help us to cure the cancer. Now, what actually disturbs me is that the Globocon predicts that the number of cancer cases will increase to a huge number, 28.4 million in 2040. And yes, it is prepared with a thoughtfulness, and yes, they have considered that by 2040, we would have a higher medical advancement. They are considering that. But even after the medical advancement, cancer is going to hit 28.4 million people. So where are we lacking? Maybe these reports are considering that the drug discovery route, the routine drug discovery route where a small molecule from the lab reaches to the doctor's prescription, and it takes a huge amount of time for around 10 to 15 years. So along with that, this drug discovery route from the lab, from the bedside to the bedside, it also needs a lot of money associated with a lot of cost, associated with time, and as well as it is associated with the frustration of the scientists. Why I'm saying so? Because in lab, when we start with, suppose with 10 molecules, then with time, at times it happens that we end up after the clinical trials and all, we end up with nothing. So it is associated with a lot of frustration as well. So do we have any option with this? Yes, we do have an option. We have the traditional health medicine system from which we can take help. We can impart the reverse pharmacology where we can utilize the knowledge which is there lying in the traditional health system, in the ethnopharmacology. We already know few of the plant extracts, we already know few of the formulations that are prescribed by the experts of these traditional health medicines. They are prescribing them. So let us utilize that information and let us move back, let us reverse, and from the bedside, let us move back to the bedside and identify the specific molecules from that information. Patanjali is sort of following that route as well, and I would say that we are performing a very systematic research. Why I'm saying so? Because we have a specific protocol how we are moving towards and how we are following this reverse pharmacology. The first step is the development of the formulations, and these formulations are inspired by Ayurveda. Once these formulations are prepared, then we have a very strong herbal and analytical chemistry team which actually properly characterizes these formulations. These characterizations actually help us to identify the specific phytometabolites, bioactive molecules, so that we already have an idea that these phytometabolites could have a potential against cancer. And then the next step goes to the lab where through in vitro experiments, ex vivo, in vivo experiments, or the microbiological experiments, we actually identify the mechanism of action of those herbal extracts, those formulations, and we identify the bioactive molecules which are present here. So today discussing about leukemia and lung cancer research which we are doing here. So since we have a very broad range of audience here which are coming from different backgrounds, so I would keep my presentation very simple. So if I talk about leukemia, leukemia is in a layman language, it is blood cancer. So let us understand what is the function, what actually blood do. If I talk about the composition of blood, it includes plasma that gives the matrix to the blood and the formed elements. Formed elements are the cells. So this yellow part is the matrix and these are the cells which are present in the blood, right? So these are the formed elements. This plasma helps in the transportation of the nutrients, electrolytes, salts to the whole body, and these formed elements impart the immunity to the individual. So these are the functions of blood. Now what would happen if these cells doesn't perform their function? So let us understand how these cells are formed. This is a hematopoietic process where a hematopoietic stem cell converts into precursor cells, and these precursor cells become red blood cells, granulocytes, macrophages, megakaryocytes, platelets. So they have a destination, right? So what happens in leukemia is that this conversion from the precursor cells to the specific cell lineage, this gets perturbed. And what would happen is that this is a normal blood where you have properly functional cells which are lying there. We have RBCs and different type of cells here. But in case of leukemia, the blood cancer, what would happen is that we have RBCs. Yes, we have few cells. We have some specialized cells. But in addition to that, we have some precursor cells. These we call as blast, and these are just lying there. ऐसे के ऐसे रहती हैं, proliferate होती रहती हैं, but function कुछ नहीं है इनका. ना immunity impart करेंगी, ना inflammation induce करेंगी, बस ऐसे ही proliferate होती रहेंगी. So that is a serious condition, and this is blood cancer. So as I said that our first step, we move ahead with very systematically. So the first step is the development of the formulation. So the formulation which I would be talking about, which we have tested against leukemia, is Divya Cystogrid Diamond. This is a unique formulation. Why I'm saying so? Because in addition to the herbal extracts with bhasmas, we have used Hirak Bhasma in this. Now, Hirak Bhasma is a diamond, right? So we are not using it as a metal in this formulation. Rather, this diamond, these are the ashes of diamond, which are highly, which are processed through a lot of steps, and then they are highly processed through the process that has been described in Ayurveda. So we are using that diamond, those Hirak Bhasmas. And why we have used that? Because in Ayurveda, in the ancient manuscripts, Hirak Bhasma has been called as Mrityunjay, something that has the quality to defeat death or delay the death. So we believe that Divya Cystogrid diamond do have potential of hitting leukemia. So we started our research with this. So how are we going to test this? So for leukemia in our labs, we have a few cell lines which we use for testing, and these cell lines, once upon a time, they were taken from the patients, from the leukemic patients, and then they were propagated in the cell culture, in the labs, right? So what we did was, this is a K562 cell line, which is a type of leukemia, which is chronic myeloid leukemia. So in these cell lines, we have treated Divya Cystogrid diamond, and this is the bright field microscopic images. This is how cells look like, which are untreated. Untreated means we have not given Divya Cystogrid diamond here. Now, when we have treated Divya Cystogrid diamond, you can clearly see that the morphology of the cells have drastically changed. When you see this dense material, this is the nuclear material which is present here, and the translucent is more of the cytoplasm. Now, when we have treated Divya Cystogrid diamond, you would see that this nuclear material has started contracting. It is condensing. It is changing shape in few of the cells have become larger. So it indicates that yes, Divya Cystogrid kuch to kar raha hai cells ko. So to be more sure, we have stained these cells. We have used a stain, that is Jimza stain, so that we can clearly see that how the nuclei look like and how the cytoplasm look like. Okay, so these are the untreated cells, and you can see that there's a big nucleus which is lying there in these cells. But when we have treated it with Divya Cystogrid diamond, it is clearly evident that the nuclear has changed its shape. It is changing its shape, right? That means it is inducing something. It is trying to convert the blast into specific lineage. Now, this is another cell line, right? This is HL60, which is AML, acute myeloid leukemic cell lines. This is another different kind of leukemia. So we have tested Divya Cystogrid diamond in that leukemic cell line as well. Here also you can see that this is untreated cells, and with Divya Cystogrid diamond treatment, the morphology has drastically changed. The nuclear material is getting condensed, and if we are going to stain them, yes, it is the nuclear material, there's no doubt that the nuclear material is changing its shape, and if you see that the morphology of the nuclei is drastically changing. So what does these morphologies indicate? Now, if I'll go back and if I'll compare this image with these images which we have got in our lab, we'll see that the nuclear morphology looks like these cells, right? So they were blast, they were having a huge nuclei. With the treatment, they are changing their morphology. Now, how are we going to identify that what is this morphology? Ye kaun sa cell bana raha hai? Ye to bahut sari cells hain. So what we are going to see is that every cell is going to express a specific surface marker. That is a protein. ये वाला सेल एक अलग टाइप का मार्कर प्रेजेंट करेगा, ये वाला सेल एक अलग टाइप का मार्कर प्रेजेंट करेगा. हर सेल की अपनी एक आइडेंटिटी है. So what we are going to do is we are going to stain these cells, and we are going to see that if we test around 10,000 cells, 20,000 cells, so our aim is to see कि उस 20,000, 10,000 cells के अंदर में से कितना percentage of the cells हमें ये वाली morphology दिखा रहा है. ये वाली morphology दिखा रहा है plus कौन सी cell type की morphology दिखा रहा है. So we have used a technique that is flow cytometer, which is present in our lab. So we have passed these cells after the treatment with Divya Cystogrid diamond with that flow cytometer, and we have seen that this is an untreated one, and this is CD41 marker. As I said, this is a surface marker. CD41 is a marker of a megakaryocyte. Megakaryocyte वो cells जो platelets बनाती हैं, ठीक है? तो हम देख सकते हैं कि untreated में ये वाली जो population है वो 18.8% है, ठीक है? Now with Divya Cystogrid diamond, this population has increased from 18.8% to 42%, and there is no doubt that Divya Cystogrid diamond is actually effective against leukemia. This is the surface marker, and then we have looked at that the gene levels are also increasing for the same protein. Now this is another marker, CD11B, that is also increasing from 8.6% to 17.92%. Now this is K562 cells, and if we come to the HL60 cell, another leukemic cell line, now you see this is a hump which shows 27% cell lines are untreated, but with the viscistogrid diamond treatment, this hump has actually increased, which shows that the population, the percent of the population is moving and showing positivity towards CD11B expression levels, and which has increased from 27% to 43%. So what is the way forward to this? We have a lot of work to do now. First, with the preliminary experiments, we conclude that the viscistogrid diamond, the mechanism of action is actually lying in the differentiation therapy. What is differentiation therapy here? As I said, in leukemia, this process from precursor cell to conversion of the specific cell lines, this process gets perturbed. But with the viscistogrid diamond, this process is again happening, right? So this is how it is happening. But still we have to dig in more mechanism of action in detail so that we understand that what is the exact mechanism of action of the viscistogrid diamond. And the next step is going to be the patient samples. We are going to take the patient samples and we are going to treat them with the viscistogrid diamond, and we'll see that what is the percent of the population, the blast population that is not doing anything in the patient, what is the percent of the population which is again becoming functional. Now coming to the lung cancer project, we have Pistecia antigerima. Now this is an example. Patanjali is not only preparing complex formulations, but we are also looking into the formulations which are developed from the single herb. And this herb is Pistecia antigerima kakrasinghi. In Sanskrit it is known as karkatashringi. And as I said, that our research, our formulations are inspired by Ayurveda. In Ayurveda, this karkatashringi, kakrasinghi, Pistecia antigerima, it is shown to have potential against the respiratory disorders. So we decided to look its efficacy against lung cancer cells. This is here what we have done is that this is the Pistecia antigerima gall. We have prepared the extract. We have treated the cells with lung cancer cells. Now when we are treating the lung cancer cells, we can see that the cells are moving towards apoptosis. Apoptosis means cells are dying. वो cells को मार रहा है. And we have chalked out the specific mechanism of action, how it is killing the cells. It is upregulating and activating specific proteins. We have identified those proteins, and then because of the activation of STAT3, ERK, phospho-AMPK, it is inducing exaggerated autophagy. Autophagy means cells को खुद ब खुद खाना. अब अगर वो process बहुत ज़्यादा हो जाता है, so cells are moving towards apoptosis. So this is the mechanism which we have identified. So along with that, we have taken help from our herbal chemistry and analytical chemistry team, and we have identified the specific phytochemical which is responsible and majorly imparting this anti-cancer potential in Pistecia antigerima. And this is penta-O-galeol beta-D-glucose. We have identified that this is the phytochemical which is responsible for that. This study has been published in Frontiers in Pharmacology, and if you want to go read it in detail, we can just go ahead and read this. But we have not stopped here. We are still working on Pistecia antigerima because we believe that there is a lot more of mechanism of action which is associated with this. So we have just treated the cells, the lung cancer cells with Pistecia antigerima, and subjected that to a high throughput study, that is RNA-seq, and looked at the treatment is actually modulating thousands of genes, and in those thousands of genes, it is the majority of the genes are from the pathways of cell division, autophagy, apoptosis, cell migration and invasion. So ये सारे pathways हैं जो कि एक cancer cell के अंदर perturbed होते हैं, सही से function नहीं करते हैं. So हमने देखा है कि जब हम Pistecia antigerima का treatment कर रहे हैं, तो इन सारे pathways की genes को वो affect कर रहा है. मतलब वो कोशिश कर रहा है कि वापस से cells normal हो जाएं. And not only this, we have identified the genes that are specifically, like these are the broad pathways, but we have identified the genes that are specifically, like in the cell division, it is from metaphase to anaphase conversion, and we are still working on this. And hopefully in the next meeting, we would be in plans to patient series 4, we would be sharing more data related to this. That's it. And in the last, I would like to thank Acharya ji and Swami ji for providing us the infrastructure and the facility to perform these research. Thank you.
Thank you very much, Dr. Savita. So that was the last lecture for today's session. Now we will proceed पधारे और सभी speakers को momento प्रदान करके उनको सम्मानित करें. May I invite upon stage Professor Xian Zhang, Professor Sandeep Bharate, Professor Anurag Srivastav, sir, Professor Partha Roy, Dr. Rajesh Mishra, and Dr. Savita Locha. परम श्रद्धेय श्री आचार्य जी महाराज, आपसे मैं सादर निवेदन करती हूँ कि आप कृपया मंच पर ही रहें. हम पैनल डिस्कशन के लिए प्रोसीड करने वाले हैं. सर. Thank you very much, everyone. I am sure we all must be having some takeaways from today's session. So let's break the ice and bring up the thoughts, ideas, concerns through the panel discussion on the topic, how to put ethnopharmacology on the center. May I now invite our panel chairs, May I invite our panel chairs, Professor Domenico Delfino, Professor C.B. Dhanraj, may I now invite our panelists, Professor Jacobus N. Elof, Professor Yuan Xian Chang, Dr. Sandeep B. Bharate, Professor Partha Roy, Dr. Rakesh K. Singh, Dr. Rakesh Maurya ji, and Dr. Anurag Srivastav, sir. I request the panelists and chairs to please be seated on their respective seats. And from here, may I now request Dr. Anurag Vashne, sir, to kindly take over and moderate the panel discussion.
So good afternoon, everyone. Good evening, everyone, and very soon going to be good night as well. All of you have been very kind, patient, and full of enthusiasm during the whole day. So thank you very much indeed. And just to ignite yourself, you could clap for yourself as hard as you can. Thank you. So we are in the last phase of the day today. Just after this panel discussion, we will move towards a little bit of entertainment. We try to see what happens when we marry yoga with music. That is just coming after this panel discussion. So the panel discussion that we are going to have it today, and I'm going to serve as a moderator, trying to find out that how to put ethnopharmacology at the forefront, and that's the theme together is. Today's chairperson for this interaction is Dr. Dhanraj sir and Dr. Delfino sir. So I think we need to start hearing from one and another in a very short window. Like, let's have everyone's thought about how to put ethnopharmacology ahead. And maybe I would request all of the distinguished speakers and the panelists sitting over here just to give like 2-3 lines of their view that what can be done, how it can be done, and what are the lacunae. And after that, we can open up the floor for the audience and if they have any questions. If they don't, then maybe I have some that I got from audience as well. So let's start maybe with Professor Dhanraj.
His Excellency Swamiji, His Holiness Acharya-ji, esteemed dignitaries on the dash, respected professors, honorable faculty members, and my dear brothers and sisters, it's really an auspicious occasion for us to dig out the ancient green gold that is ethnopharmacology. This green gold has been hidden inside, readily prepared by our ancestors. And it's time for us to explore this, bring forward for the welfare of the humanity, especially the ailing population of mankind. We have several diseases which do not have answer yet. We understand from the morning talk, several learned professors have expressed the major gaps in the present system where we are not able to understand how to control the gene mutations, how to control these various metabolic disorders. And for this, the great work what other people are doing in the different parts, I really appreciate going forward, especially the process how it has been done in various medicinal herbs. For example, Professor Jacob Nicholas Elof has expressed today morning regarding the grapeseed, prosidine. It's an excellent medicine for cancer, the grape seeds, black grape seeds. And also he expressed the Terminalia serica, Terminalia cebula, Terminalia arjuna, same family. And Terminalia serica is a phytobiotic and found to be better than gentamicin. Appreciable, because in my practice I've seen one patient, not one patient, it was my lifetime's experience as a young doctor. I was working in a hospital. A young person who has come from abroad was given, for urinary tract infection, was given gentamicin without a test dose. It was a big mission hospital with all the specialists around. I was a young doctor there just looking after things. My friends, I want to share with you the young person collapsed and died because of aminoglycoside reaction. So in such scenarios, the Terminalia serica, definitely the best answer for gram-negative bacteria. That's what has been done. So we are really going forward, bringing ethnopharmacology to the mankind. And Sitesh Bachar has really expressed several Unani drugs. And our great Y.S. Chang, I really appreciate him for the standardization of these various herbal medicines, especially aflatoxin levels and TCM herbs, the process of standardization, etc. And the way Dr. Rajesh Mishra has put forward the 20 herbs is a gift for the mankind. The way he shows our taki, just like intestine, Jyotishmati, just like brain, which is acting on them, I really appreciate Rajesh Mishra's presentation. And we have Sandeep B. Bharta from Jammu for unification of Ayurveda, Unani, naturopathy, to go for the biomolecules. This is the way I said that rethinking of phytomedicine to take forward, his suggestion is really great. And that's why you get the clearance from FDA. Yes, it's a time for us to pull our socks, reunite, and see that how best we take forward the phytomedicine. And next is, we have this wonderful presentation by the great Anurag Srivastava-ji, who is the instrumental for really putting the face of plants to patients. This is a brainchild of Dr. Anurag Varshine and as well as Dr. Anurag Srivastava. Both Anurag synonyms, Anurag Square, has brought this great jolt for ethnopharmacology globally. Please give them a big applause. We bow down to you, sirs. Yes, and Partha Roy really expressed regarding Resveratrol in grapes, isothiocyanate from and pterostilbene. Fantastic, they have really identified the molecules to control the apoptosis, to see the apoptosis pathway and its inhibition. They all act as anti-proliferative, anti-metastatic. Fantastic, fantastic way forward for ethnopharmacology. And what Dr. Anurag Varshine and his team is doing, that is Dr. Savita Locha, she's really has opened the new doors of working on HS60 cells for leukemia, cystogrid, diamond. From Rashastra, the diamond basma was being neglected by several pharmacologists globally. I want to bring to your kind notice, when I started my graduation, finished my graduation in 1984, I was hated to use Hirak Basma, diamond Basma, because we do not have tools to say that Hirak Basma controls the proliferation of the leukemia cells. Today they have opened a new doors for ethnopharmacology. And the great work done by our Savita Halder on Anuthaila. As an Ayurvedic physician, I bow down to her for its work on mycosis. So many people died. Had we done it before, about 10 years, 5 years before, had our governments rose from the slumber to activate this ancient system, so many lives would have been saved. And the great work goes, the credit to our Swamiji and Acharji to bring the great people like Anurag Varshine on the board and Savita Halder on the board to open up this special research. Please give them a big applause, please. And I appreciate Savita Locha's work, especially on Pistacia integrima. It is also called as, we used to call as Ras succedaneum in those days. It's now called as Pistacia integrifolia, that is Karkatashrangi. They identified this chemical, penta-O-galoil and beta-D-glucose. This is the one which is stopping the apoptosis and autophagy of the cells in lung cancer. So I really feel that the great treasures of Ayurveda expressed by Bhava Mishra, Charaka, if these herbs are taken up by the great people of here globally, and if we all really take one plant or one disease mentioned in the ancient texts and work on it. Let us not really put all the burden on the Patanjali and Dr. Anurag Varshine and his team. I wish we all take up for the clinical trials as our dynamic leader, Dr. Anurag Varshine, told, reverse path. Man to the molecule. It's a reverse path. We assure you that the medicines mentioned are safe. Ask your governments to go for the clinical trials. This is what I'm proposing. Clinical trials that have a collaboration globally. Clinical trials of herbal medicines. One body, and we do it globally at different centers on the same disease, and see how the humanity in different countries, different climates responds to the particular disease. I really request that this be put it in one of the agenda, and request all the great people to come forward to make an international collaboration with Patanjali Research Institute and start the clinical trials.
Sir,
with this, I sincerely thank the great leaders giving me the opportunity to speak on this. I'm really still a chair, and shall try to give my comments from time to time. Thank you so much.
Let's have a big round of applause for sir for summarizing the whole day that we have been through, and that has made the next part much easier, because we are planning to go for the day summary, and all of you have already heard it now from sir, that what has been discussed since morning and how we have taken ahead. So now let's come back to the panel discussion, and we are talking about how to put ethnopharmacology on the center stage, and what are the views for all the panelists sitting it right over here. Sir, thank you very much for putting the summary in such a way that we can now start having discussion. So I think I made an error that I jumped the queue. I should have been first gone to Professor Partha Roy, and he was elbowing me saying that why you are not asking me. So I'm correcting myself and giving it to him so that we hear from him, and then we move to the next one. Dr. Roy.
Okay, thank you very much, all the panelists, and of course, to Anurag. So I think I heard the whole day's deliberation, and I understand the whole next 2 days events also. So these things are moving in a very, very encouraging and right direction. And what I feel that the way forward is reverse pharmacology, as Dr. Anurag ignited in the morning itself. So reverse pharmacology is a way of authentication of the existing mode of herb-based therapies or traditional knowledge system. So if we do this authentication process using proper higher-end molecular techniques like barcoding and all, understanding the mechanism of action, because ethnopharmacology is very, very critical, because most of the time we target these phytochemicals targets multiple targets, multi-targeted approach. So naturally, as much as we can utilize its targets, one single molecule can target multiple sites, multiple organ, multiple tissues. The more we explore that, we can better understand its mode of action. So single drug can cure multiple diseases, multiple symptoms. So I think more science has to be done. So as we are doing it out, all this as Patanjali research institutes, whole of Patanjali groups are doing. So we should all come together. Let's exchange more ideas, and authentication, taking it forward is more critical and in need of the hours, and they are really doing it. I think this should be continued further, and we should be part of its system, and we can use it. Thank you very much.
Professor Delfino, the co-chair of this panel discussion, how about your views and the guidelines for us to move ahead? You can try this mic.
Okay. First of all, thank you very much. I would like to express my gratitude for this invitation. I could not expect this young audience, and I am really happy for this. So I want to give an applause to the young student. Thank you. Well, how to put ethnopharmacology on the center of the stage? I think you don't have here this problem, because in India and in general in the Far East, really the ethnopharmacology is in the center of the stage, and this is further boosted by the integration with the allopathic medicine and with the new biotechnologies. I think this integration is fundamental to keep the Ayurvedic medicine and the ethnopharmacology in the center of the stage. The integration is always important. The cooperation is always important, and only only through this integration, this cooperation, everything can be improved. Maybe the problem is with the Western world, with my world, Europe. I come from Italy, where the ethnopharmacology almost does not exist, because the herbal, medicinal herbal heritage was destroyed during the centuries. In the Middle Age in Europe, the medicine was based on three stones, which were drugs, surgery, and diet. We almost always eliminated the diet and the drugs as herbal drugs. But now, in the last 20, 30 years, herbal drugs are coming up again, because the confidence to the allopathic medicine was going down in some way. And how can I say, what really I wish is this new fortune of herbal medicine in Europe, will continue to grow and to integrate with the biotechnologies, with the Western medicine, in order to move the balance from the disease to the prevention of the disease, to the lifestyles, which we don't consider too much in Europe. So this is my wish. I mean, for example, yoga is very popular now in Europe. I myself do some yoga. Yeah.
You will do tomorrow as well.
Yeah, I do. But it's considered an exercise, not a medical system, yes. So what I wish is that some of the concepts of Ayurveda medicine can be transferred to Europe for improving the health consciousness of the people. Okay, thank you.
Thank you very much, sir. Professor Chang, we would like to hear from you as well what need to be done to put ethnopharmacology on the center stage as the main treatment option for the patients and for healthy people as well.
Thank you. I'd like to thank you again for the invitation to participate in such a great event. The main theme today, we talk about ethnopharmacology. We understand ethnopharmacology is the human experience on the use of the herbs. As you understand, in early days, when those Western pharmaceutical companies try to invent new drugs from natural sources, they may do random screening, try to collect 5,000 or 10,000 herbs from different areas of the herb, and then try to do biological screening. And then it end up, they spend a lot, great effort, but end up with very little. So therefore, what they did was, based on particularly my professor, Norman Alfonso, in early days in Chicago, he had a database. And then based on the database, he had ethnopharmacology use from different sources. So if they, for example, if they try to do lowering blood pressure, try to develop blood sugar lowering agent, then they try to search around. If there's a herb that had been used in one particular area, person, it's not enough. But for the same herb that was used in different area, unrelated area, and all claim that it has can low blood pressure, blood sugar, then it's very likely it's going to get positive result. Okay?
So of course.
So that ethnopharmacology is very important. And secondly, I'd like to emphasize, of course, Chinese medicine we regard as ethnopharmacology, and Ayurveda medicine in India also are ethnopharmacology. And I'd like to say, even though we use some herb traditionally, yes, in Ayurveda medicine you use very good, and in Chinese we use, but without any evidence, any nowadays scientific evidence. So we try to do studies on our traditional Ayurveda medicine or Chinese medicine and try to have a scientific proof. And then we can feedback those results to the Ayurvedic community, to teach Chinese medicine community, so they feel more confident when you use the same herbs. Okay? And lastly, I like to echo the term you used in the presentation, reverse pharmacology. Okay? I'd just like to give an example on the TCM herb to fight against the COVID-19. They, based on a clinical formula prescribed by one of the TCM physicians, and then they give the medicine directly to the COVID-19 positive patient. And he said, it's getting very well.
Of course.
So based on that, then they start doing some basic study, so on and so on, and then become, more people feel it's very good. And then they turn license out to the pharmaceutical company. So I think, without that, they start to do basic study and then try to come up with a new drug to fight COVID-19. No way, okay?
Of course.
So I think that's my opinion. Thank you.
Thank you very much, sir. Professor Ilov, your views, please.
Thank you very much for the opportunity to talk about this. I agree with what the other people have said. Reverse pharmacology is the answer. So there are some steps. You look at the old information and you select certain plant species and you test whether the activity that has been ascribed is true. And once you do that, then you go and isolate the active compounds. So you use the information that comes from many, many years, but you test the information as well. I know people that, or I think, let's put it that way, people that use plants thousands and thousands of years ago were as intelligent as we are. There were no difference in intelligence for people, but there were a lot of difference in knowledge. Certain things they knew which we do not know. But so what I would just add, I think you are going the right way. Because if I see the way that you handle things, it's exactly right. Maybe I can just add one more thing. If I talk on behalf of Africa. Africa contains 1/3 of the world's plant species, 1/3. And with the exception of a little bit of information in Egypt, there's hardly any information in Southern Africa, or Central Africa, East and West Africa. So the way that that is handled, people go and ask people which plants are used for which indications. And there's the Journal of Ethnopharmacology published a lot of that information. So that brings a new way of looking at things. And finally, I'm impressed by the excellent work done by everybody. And boy, I would have loved to work in this institute.
Absolutely,
but
sir.
Just one more point. I think what you should also consider, let's say in Ayurveda, a certain plant species has been used. Look at other related plant species, because the development of the species through evolution runs along chemical lines. So the chances are very good. If you find activity, you may find a related compound without having to do the extra work. And then you can test that. But congratulations, I'm impressed with what you guys are doing. I don't think you have to rethink. You're doing it exactly right.
Thank you very much indeed, sir. Thank you very much for those advices. Professor Maurya.
Good evening, everyone. I'm grateful to Param Puj Swami ji, Acharya ji, and Dr. Anulom-Vilom for giving me the opportunity to participate in this wonderful conference. I enjoyed all the lectures from morning to evening. Elegant presentations. But one of the speakers cited that very good example that 80% of the world's population rely on the traditional system of medicine for their primary health care. So I want to mention here that traditional systems have been the starting point for the discovery of many important modern drugs. Take the example, very simplest example, everyone are using this aspirin. This was isolated from Salix alba, and whole world is utilizing. So this title, Plant to the Patient, is very well chosen for this conference. Plants provides treatment for patients. So one thing I was thinking that we also need to look the plants, need to find the plants which has not been worked out so far in order to find out new biological activity, new bioactive markers, because these plants provide structurally different molecules. So there is still need to find out the new plants. It is also mentioned in ethnopharmacology, also, ethnic medicine also, that those plants have not been worked. For example, like Almus wallichiana, this is mentioned as endangered plant, but when we look the literature, nothing was reported in this. Only one thing was reported, that stem bark of this, if it is applied to the fracture place, and heals the fracture. And when we look to the chemistry, there was no report, nothing in the literature. We isolated novel molecules from this. Almus wallichiana, this is grown in this area only. So we need to find out such types of plant. And thank you very much. This is my...
Thank you. Dr. Bharate, your pulse on wisdom.
Yeah. Thank you for this another opportunity. So rethinking ethnopharmacology, I think just kind of changing the perspective of the end user. So people think that like traditional medicine or ancient medicine, so they are not really well accepted. Still I'm saying it in a different way, because everyone knows that old is a gold, but we have to prove that, I think there is a need of a scientific validation. And if one has to do the scientific validation, there are, I think, 3-4 things. There is a path has been created by a regulator, we have to follow the path. So first thing is select a right plant and choose a right disease for that. So once you do that, you know that there is a efficacy, there is a safety is there because it is being used since ancient times. You have to forget that and start from the scratch, do a robust naturopath chemistry, isolate the pure compounds, test each and every compound for the biological activity, so build a confidence that this is the compound, it is working, so my extract should work. There is no any confusion about that. Once you establish the chemistry, then efficacy is there. You know efficacy is there, you have to prove that efficacy again in the scientific models. Once you prove the in vitro, in vivo efficacy, then obviously you have to do the regulatory toxicology so that you can convert the traditional medicine tag to the modern medicine. Once you convert that tag, then you are into the game. And I believe that Patanjali has created everything, right facilities are there, right people are there. Only you have to follow the path to make that traditional medicine to the modern medicine. Thank you.
Thank you, sir. Dr. Rakesh Singh.
So, like in one of the presentation of the speaker, we got to know, in spite of having so much of traditional knowledge about ethnopharmacology, still we do not have any NDA filing or IND filing. It was surprising to know, probably all of you might have been surprised by this, right? Only 2 IND filing and it got approved. So why we are not moving ahead towards either NDA filing or IND filing is due to lack of evidence, scientific evidence. So in my opinion, probably there are 3 things that we need to do. One is to back up the traditional knowledge with scientific evidence that we can generate, any piece of in vitro, in vivo data that we can generate. Second is their uses are already known in traditional medicine. So can we repurpose them for anything else? So drug repurposing is another area for natural products. And the third thing is the major problem with these kind of agents is their delivery, their absorption in the body. So how can we work on their delivery? Probably if we can look upon these 3 aspects, I think we can move ahead brilliantly on this. And as Dr. Anurag showed in his presentation, there are 9 institutes affiliated to Patanjali. And probably all these 9 institutes are having the same vision and mission, what I'm saying. So all the best to you, and we are there with you to support all of us to move ahead.
Thank you, thank you, thank you. Now let's hear from Professor Anurag Srivastava, sir, that how do we put ethnopharmacology on center stage. Sir just joined because he was looking after a patient. So let's hear from him, and then we have questions from the audience, if at all, and any suggestions, advices, and queries if you have on the same issue. Sir, please.
Thank you. Namaskar. It's really very impressive to see that very distinguished, experienced, skilled scientists are now doing cutting-edge research to identify what is the active molecule or what is the active substance which is available in the various plants and other herbs and other basic resources described in ancient wisdom of Ayurveda, Unani and other ancient texts of herbal medicine or traditional medicine. For identifying the mechanism of action and explaining how the drug is working, it is fine. But when it comes to treating patients, we should be cognizant of the fact that the traditional wisdom of Ayurveda, as described in Charak Samhita, Shushra Samhita, and all the ancient treatises, texts, actually describes the whole herbs rather than the active ingredient of it. I shall give the example of haldi, curcuma longa. It has been described as strong antiseptic, for healing agent, pain relieving, inflammation relieving, as well as anti-cancer. So one scientist, Dr. B.B. Agrawal in MD Anderson Hospital in Texas, took the task of finding the active ingredient called curcumin. And he did a lot of research, for almost 20 years. He found that how it suppresses the NF-kappa B and all the mechanisms. But when this curcumin was brought to the patient, we found that it was not working in most of the diseases. So he promised that it will work for prevention of cancer, leukoplakia, submucous fibrosis, breast cancer, colon cancer, stomach. So there were many trials, including in the All India Institute. So as high a dose of 8 grams of curcumin were administered, and still there was no effect or minimal effect. Then he suggested add anti-cancer drugs, gemcitabine, with curcumin to have its effect on gallbladder cancer. Well, that is not Ayurveda, So what I am trying to say, then it was found that curcumin has very poor bioavailability. Very little of curcumin is actually absorbed, 1%. But when Rishi Charak described it, then he did not mean to extract curcumin. He described the whole haldi. So we should not forget the basic fact that the Charak Samhita, Shushru Samhita and all other treatises in other parts of the globe, Chinese medicine, Mexican medicine, African medicine, actually have been practicing for centuries the whole herbal or whole some metallic extracts or other salts in their whole form. And the one reason I've been told by learned Ayurvedachar is that the entire plant contains both the active ingredient, maybe curcumin or some other antioxidant, but it also contains other molecules which enhance the effect of that drug or which suppress the harmful effects of that drug or which render it easily absorbable. So हल्दी आप खाते हैं, सब लोग खाते हैं, हम लोग हल्दी अपने ही मदर सब्जियों में डालती हैं, फल में जो पौधों में दूध में डालते हैं, तो वो जल्दी absorb हो जाता है. लेकिन curcumin जब दिया गया तो नहीं absorb हुआ. तो we may be using the science wrongly if we keep trying to extract every molecule. For explaining the mechanism of action, it is fine, right? For supporting that how reserpine was working from Rolfia serpentina, that it is acting on, as a vasodilator, causing suppression of the, reduction, hypertension, and so on. For teaching in the classroom and for explaining the mechanism, that is fine. Extract the reserpine from Rolfia. But when you are going to use in the patient, and if you want to use traditional wisdom of Ayurveda and other systems of Unani and Chinese medicine, please be cognizant of the fact that it was described as being used as full herb or entire herb, right? Because the Rishi Muni had done trials, including randomized trials in those days. So if we want to do this extraction, then we'll have to do another step. That is, do a randomized trial in which in one group you will use haldi as the whole plant extract. On the other randomized trial in the other arm, you use curcumin, the active molecule that your lab has suggested. And then see the difference. See the difference in the efficacy as well as the side effects, absorbability, bioavailability, and all that. So unless that randomized trial is done, just merely extracting the active ingredient from a substance based on cell biology or animal models would not be sufficient. We'll have to do randomized trials, so without which we cannot say that this is the active ingredient from a particular, Hirak bhasm or gold bhasm or any, metal or any plant, and then make a preparation and send it to market. No, we'll have to first do phase 3 trials, as we call it in randomized trial. Phase 3 randomized trials have to be done to show that the preparation is, number 1, safe, and number 2, effective. Both safety and efficacy have to be demonstrated in phase 3 randomized trials, where you give the active ingredient derived from the knowledge of your research, basic science research, and the other group use the traditional drug as described in Charak Samhita. And then if you demonstrate that this active ingredient, if given alone, is more effective and safer, certainly, please, by all means, do you use it. But you will have to do randomized trials to show that the active ingredient is more effective and safer than the whole herbal extract. So unless that is done, we should not be sitting quietly and not clap ourselves and not, right? So I think we have to be cognizant of this fact. Yeah. Yeah, thank you.
Thank you very much, sir.
I appreciate Dr. Anurag Srivastav's decision, but there are two versions here. One is our doctor said that go for the active principle and go into the profiling of that particular active principle. And other version is go to the randomized control studies of each of these substances of that active principle. I really feel that Anurag Srivastav, what he said is good, but let us all agree now to that, or shall we go forward? Because it is a panel discussion, it's open to the forum to decide. This has been always a controversy since my last 2.5, 3 decades scenario of scientific practice. The first failure was Ravolfera serpentina regarding the serpentine, and one was success was in the hyoscine methyl bromide we use as antispasmodic, and it is really successful, but Ravolfera serpentina failed because it had some certain problems. So this is this scenario, this is the issue to be open to everybody to decide to go for active principle 1 or whole of the medicine. We have honorable principle here also. We all should agree to that point that we go for a whole medicine as prescribed in the text, is what I find.
So basically I think what this debate is going on, discussion is going on, I must say, both are important. In our laboratory, we have taken 3-4 examples. One example is black pepper. Black pepper consists of piperine. We have tested in cell-based system, animal-based system for its anti-leukemic properties. And there was a difference when the whole black pepper extract was used and piperine was used. There was a difference in the activities. Whole extract was better than this one. But understanding, yeah, definitely is there. So mechanism-wise, they acted almost similarly. So that is there. We did one more experiment on betel leaf, pan patta. Betel leaf's major constituent is hydroxychavicol. Again, we tried experiments using hydroxychavicol as well as betel leaf extract. And both the time we found the mechanism almost same, efficacy of the extract was better. So of this extract was better. So that means what sir is saying is very true. So we have to go for both, we have to test both of them together. If we go to clinical trial, it will be very expensive process to try hydroxychavicol or piperine independently. If the in vitro and in vivo studies shows that mechanism is same but efficacy is high for the extract, we should directly go to the extract and formulation as traditional usages. We don't need to go for individual isolated molecules. But it is always a good idea to test them independently as well as the whole extract. So we get an idea what is really happening or what is the difference at all. I think that's a good idea.
Okay, so we have now two different approaches that we have been discussing, and which is very nice and healthy. But I would like to add one point, what we heard from Dr. Bharate. I think what he meant to say, he's not saying that a single plant extract would have a single compound that would show an efficacy. But when we're talking about the evidence, that means that the whole extract works, we have to show the which pathway it is following in the cell, because that is the fundamental unit of life. So whatever going to happen at a cellular level, eventually will go at the level of tissue, at the level of system, at the level of whole organism. So if you know what compounds are there, it will help you to find out that the pathway we are going to go through, the evidence that we are trying to generate. As such, if you take the entire extract, it might become very difficult. And I'm sure Dr. Bharate can correct me if I'm saying it in a different way. But the thought there was to actually generate the body of evidence, which really rethinking ethnopharmacology, to find out that what are the evidence we have it. Of course, clinical evidence is the top priority. It trumps everything else. But as Dr. Roy just said, it is super expensive for doing 20,000 trials. So you need to be selective, and you need to be looking at what might work better. So the guidance from in vitro biology and in vivo biology would be more useful. That's what I think I would like to add.
So I'll just add to this.
I think the point made that there could be synergy within an extract is of course a very good point. I mean, there are certain antibiotics that contain two different antibiotics, then it's much more powerful. But one thing I must mention, the terrible quality control problems that you have, variation in plant extracts, plant grown in one area have different chemicals than a plant grown in another area. So the situation with using an extract is much more complex than if a single compound was active. If a single compound is not active, then it means you have to look and you have to find, do we need possibly 2 or 3 compounds? And it is complicated because then you must look at collections of the same species from different places, and one can do it with metabolomics, I think, and then you can see you need actually these 3 components in a certain ratio. So while I think we have a tremendous benefit with the ancient knowledge, because people have worked many, many years on that, I think we now have the apparatus and the intelligence to do something that you can have a very high predictive value that it will work. I don't take any medicine, really. I hate taking medicine, but I think I said enough.
Yeah, I'll just add to Dr. Vashne and Dr. Srivastava said. So I partially agree with Dr. Srivastava what said. So his opinion about sticking to the crude drug, and as far as we move from the more and more purification, we lose the connect with the traditional medicine. That is very true. But ultimately, since we have to convert the traditional medicine to the modern medicine, we have to think about the pure natural product. And for that purpose, this DCG has created an in-between tag. Like, I'm not saying that go to the pure molecule and does not stay to the extract. So in between, they identified that we make an enriched fraction. Still, it is a mixture extract. It is not a pure compound, but it is enriched fraction. And that way, you link to the traditional medicine and you talk about the pure compound. We know that in this fraction, this is the compound which is responsible. And that way only we can link this traditional medicine to the modern medicine. This is my...
That's actually a very valid point.
Yeah.
So I think let's hear someone from audience. If they have any queries and questions and suggestions, please, I am seeing a big hand is being raised. So please ask if anyone else from PRI or from university or from Ayurvedic college. Each question is a good question, and there is not a single stupid question that ever came on this earth. So do not hold yourself. This is a good opportunity. Please do ask openly and frankly. If you get the good answer, you will be enriched. If you don't ask a question, you remain that way then. So please ask.
Yeah, Mike.
We should learn from very experienced Ayurvedic physician, our principal of the Patanjali Medical College, Professor Anil Kumar, and Dr. Suman.
Sir, can I speak in sitting condition? Okay, sir. Thank you. So मैं हिंदी में ही कहना चाहूँगा कि जो अपनी बात है उसको अच्छी तरह से आप पैनल में डिस्कस करना। दरअसल जब मैंने अपना पीजी में कर रहा था तो उस समय मैं रुड़की में इंजीनियरिंग कॉलेज में गया था अपनी ड्रग्स को लेके। तो उन्होंने मुझसे एक बात कही थी कि अगर आप कोई भी ड्रग ले रहे हैं, लेने के बाद में आपके जो जितने भी एंजाइम्स हैं वो उसके साथ मिक्सअप हो रहे हैं, जितने आप जो रिएक्शन करता हार्मोन्स के साथ करेगा, एंजाइम्स के साथ में करेगा, उसके बाद में उसमें क्या चेंज होता है, जो एब्जॉर्प्शन होता है, एब्जॉर्प्शन तो बाद में हुआ। अगर हम इन विट्रो और इन विवो वाली स्टडी करते हैं तो वहाँ पे कोई भी उसके साथ एंजाइम्स वगैरह नहीं मिल रहे हैं। तो क्या इसको हमें ध्यान में जरूर रखना चाहिए? क्या उसमें कुछ चेंज होता है? बस मेरा यही एक पैनल के साथ में डिस्कशन। थैंक यू।
ओके, सभी को सर, हमारे स्वामी जी हमेशा कहते हैं दैट कैंसर सेल्स कैन नॉट थ्राइव इन ऑक्सीजनेटेड एनवायरनमेंट, right? So the query lies is can there be a possibility like we infuse oxygen at the specific points of the cancer cells area for them to do the process of apoptosis, or is there a mechanism where these cancer cells can be reduced? जैसे हम कहते हैं, स्वामी जी कह रहे थे अनुलोम-विलोम is the cheapest way to get the cancer cells decayed or removed, but अनुलोम-विलोम से तो हम वो एक लंबा procedure है, it has to go to the cells probably at the brain areas, but is there any other way where direct, वो बिल्कुल direct वहाँ पे जहाँ पे होना है वहाँ पे पहुँचे ऑक्सीजन और heal हो पाए? Yeah, thank you so much.
I have no idea.
You answer the question, please.
Very important point. So studies have shown that hypoxic cells are resistant to cancer treatment. So when we give radiotherapy, the hypoxic cells cancer cells do not respond. So they have tried to increase the oxygen concentration within the tumor by putting the patient in hyperbaric oxygen chamber, where the oxygen pressure is 2 to 3 times higher than atmospheric pressure. Atmospheric pressure is 760 millimeter of mercury, right? So you put the patient in a chamber with 100% oxygen, which is at 2 atmospheric or 3 atmospheric. So at higher pressure, you know Dalton's law of partial pressure works, and Henry's law of diffusion in physics, you must have read that. So with increased oxygen pressure, more oxygen will diffuse through our lungs and through alveoli. It will get dissolved in plasma. It will reach the cancer cells and whole rest of the body at higher concentration. There are randomized trials and there is meta-analysis, Cochrane overview, to demonstrate that at least in 2 cancers I know, in cancer of the head and neck and cancer of the cervix, patients respond better, they live longer when hyperbaric oxygen is combined with radiotherapy for cancer of the head and neck and cancer of the cervix. So there is randomized trial and Cochrane review. You can read Cochrane reviews. Cochrane review are considered the most robust form of evidence based on meta-analysis of several randomized trials, right? So at least in 2 cancers, there is strong evidence that giving hyperbaric oxygen improves the therapy of cancer of the mouth and cervix. So I think the concept that Swamiji is providing, increasing oxygen through Anulom-Vilom and other pranayam, stands to reason. Thank you.
You want to add on this? Any more questions from audience, or should we then move ahead? Oh, there we go. Ma'am, please.
Thank you so much, sir. Om, we all are very, very much honored that we are learning so much from this informative discussion of today's session. I would like to say that as Srivastava sir told that it is like curcumin, if we are talking about that it is working in synergy. So synergy like, and we are facing problem with the reproducibility of phytochemical molecules which are present in a herbal medicines. To establish reproducibility of phytochemical molecule is really a big task. If unlearned professors may give some light or on this opinion.
Thank you, Dr. Seema. I think this is one of the relevant questions with the theme of this discussion. So thanks a lot. Let's clap for the ma'am, because she had a query regarding how to put ethnopharmacology on center stage. So let's hear from the experts. I mean, how do you see the variance in the phytochemical composition from different location of the plants or the herbs?
As we have seen the presentation of Dr. Y.S. Chang, his way of standardizing the phytochemistry or phyto-herbal drugs of the place he has come, he has put the different ash values, different chemical values, etc., and ingredients, especially flavonoids, etc., etc. In the same way, we have Indian pharmacopoeia, where we have a specific levels of each herbal drug which has been mentioned. So we have been stressing even in institute also, when they are taking any herbal drug, check for those standards, whether their particular herb they are collecting for the test, does it comply with the standards prescribed in the Indian pharmacopoeia of the particular plants? If they really, after doing those tests, standardizing the drug, if they match with that, then go take that drug as a standard drug for your clinical trial. And that is what we have been doing, we have been doing at different institutes in India. I think this is sufficient for your question, madam.
Oh, she has a second question now. All right.
Actually, our pharmacopoeia, Ayurvedic pharmacopoeia, and Indian Ayurvedic pharmacopoeia, or be it Chinese traditional medicinal pharmacopoeia, they all are talking about a group of molecules like flavonoids, TPC, and all those things. We are talking about a single molecule assay and content of that assay in a traditional herb. So that, like, just a question that how we can establish a reproducibility as the plants are of different origin and different geographical locations. So they are varying for their phytochemical composition and contents.
So, okay.
Sir, 1 minute.
Please.
To continue with her question.
You please continue. Yeah, give the answer, please. Yeah.
No, I don't want to answer. I have,
Question.
the continuation of the question.
Okay.
So my thesis in MD was Tailabindu Pariksha, told by Acharya Yog Ratnakar. So the whole diagnostic or prognostic, this criteria of this Pariksha was, we had to use Tila Taila. Yog Ratnakar said, take Tila Taila. He did not specify what til, is it black or is it white. From which state it has to be taken, he did not tell. He did not specify the kind of water the patient is drinking. And I had taken that topic as a thesis topic. When I faced my Viva, the first thing I was asked, did you do the TLC of the Tila Taila? Yes, I did it with IIIM Jammu, I told. Then they said, कि आपने किसी और जगह के तिल को try किया था? So this is what the standardization is lacking in Ayurveda. I had to face 1.5 hours of Viva just because I did not knew what other things may vary in the Tila Taila composition. So they told me to take this thing, again in further study and do it in different states. We chose a land, we made someone to grow till for us under good agriculture practices because I was working with IIIM, those people helped me. But for a normal person to go for MD and take a topic from Ayurveda as a thesis topic, these things become really impossible to handle. Thank you, sir.
Can I answer this question?
Yeah, can I answer the question? Somebody answering? Yes, please.
Yeah. So regarding, I think, your question related to the how we can standardize or check the phytochemical content, particular single compound. So for that, since once you select the plant, you have to make the extract. For example, you collected from the Dehradun, one plant collected from the Western Ghats. So multiple locations, even the age of the plant also, content of the compound varies. Once we collected one plant, which is 20 years old and one is 10 years old. There is a huge difference in the phytochemical compound. So you have to collect all types of variations. You have to consider, for example, make 10 extracts. You know that what is the content of the XYZ compound. and you check for the activity, and then you specify based upon the activity, like 1% if compound is there in the extract, it still shows the activity, 0.5% it is not showing the activity. So based upon that information, you have to specify the range. So that is what phytopharmaceutical guidelines are created. Each and every four markers are there. Each marker you have to specify the range, 2 to 4%, for example. If next time you make the batch, for XYZ compound is only 1%. That batch is failed. You cannot use that batch. So that's how you have to create the ranges for each of these. And that's where the you can make the reproducible plant extracts for the development purpose.
In South Africa, some senecio species are used with very good activity. But many people are killed in hospitals because they use the wrong senecio species. You need an expert taxonomist to find out which species it really is. So there is a lot of danger if it's not a well-regulated industry, if you use an extract of a plant. So I think that's one thing that one should consider as well. Because if the values go up of the active compound and it is too high, it's more dangerous that way than if it is too low. That is why we have prescription medicine to ensure that you've got the right effective dose in the blood with the time, because it goes up and it comes down. So if the dose that you take or if the activity is too high, you can have serious negative effects. And I think that is just the answer for using a single compound, which you know exactly, but that's not the total answer. There's also difference between different people. Because some people would be more sensitive to something because the enzyme levels are higher in some people. So whenever you can get away without taking medicine, eat good food, forget the medicine.
Okay. I echo Professor's opinion, two opinions, okay? Of course, for the prime material, the same species grow in different areas. It's going to end up with variations of the active ingredient, okay? And you have been mentioned many pharmacopoeia. Here, Japanese pharmacopoeia, Ayurveda pharmacopoeia. Pharmacopoeia only set the minimum requirement. And then they have some certain variations. But that, in order to be used as medicine, at least you have to meet that minimum variation. But even though we say by growing in different area, different season, definitely it's going to have some variation. But that's not mostly important. The important thing, as Professor said, you get the wrong species. You don't get the correct species, then definitely you will not be able to expect the clinical efficacy and no way get the expected or even cause poisoning. That's it. Thank you. So the pharmacopoeia allowed a little bit variation between batch to batch, okay? But if you go beyond the average, the limit, then it will be used as medicine. Thank you.
Okay, thank you so much. I think 3 opinions have been given, and the 4th one is add to it, that we have IICT standardization. They have been doing this standardization of all of this. IICT, which is in Hyderabad, Indian Institute of Chemical Technology, will do the standardization of any drug you send. We pay the money, they give the report. And with this, I think almost all the questions have been answered. And if anybody have any more questions, please? We're running out of time. We have to relieve our guests. Yes? Any more questions? So we wrap up today now. So we have discussed important points regarding the chemical aspects, etc. And whether to go for a single chemical constituent or we take a group of them. And Dr. Anurag Varshney has given a very beautiful problem-solving method that is going for randomized controlled trials for each of the chemical molecules as a drug as a whole. And we really support our Honorable Principal's vision also. And I think with this, we try to close this panel discussion. Thank you so much for everybody. Yeah.
Thank you very much to all dignitaries for such an informative discussion session.
Good evening, sir. Good evening, sir. Sorry to excuse you.
Hello, from where, please? Yeah. Yes, sir. Go ahead.
Sir, I have actually 2 queries. First one, my first query was that in our the kitchen medicines we have, as Shweta ma'am said, that the different plants at different parts of the world or a country have a different level of phytochemicals, right? So if we have a particular amount or quantity that we have learned from our family or from our elders that this quantity with this substance can cure this particular type of disease, then if those different plants are from different places, then what would be the adequate quantity that we should decide for treatment of a particular kind of disease? And the second my query was that as sir said, that Africa has 1/3 of all the plant species of the world. So Ayurveda is a type of treatment, medical treatment that is also counted among the cheapest methods of therapies of treatment. But there are some countries that lack those kind of diversity of plants, flora. Indian subcontinent is also rich in flora. But for those countries that are not having that kind of flora or biodiversity of plants, and the cycle repeats itself that again those countries that are not rich in biodiversity would again have a high quantity of drug or a rich drug that the poor people cannot afford it. So please answer these questions.
Okay, thank you so much. 2 questions, and very briefly I answer. The first thing was to practice somebody suggested medicine on your own is really against the rule. It is quackery. We have experts and we are specialists in this area. Please consult them and use it. They are the experts in identifying and giving whether right or wrong. Kitchen medicine is kitchen medicine, but it has to be followed by a specialist. We are all kitchen medicine doctors only. We are specialists in this. We have worked out very hard to follow that. Second question was other countries' issues. We have so many people globally sitting over here. Every country has got its own specialized control system. If you say one medicine here, it's not required that they should have the same medicine over there. They got their own controlling board, and accordingly they have to practice that. So we cannot have any ruling to globally advise them unless it has been clinically and perfectly been evidence-based medicine trials has been done. Okay? The 2 things have been cleared. Any more questions, please, audience? So we close it. Thank you so much. Thanks for a great panel people and the professors over here. Thank you one and all.
Thank you very much to all the dignitaries for such an extremely informative session. So thank you, sir. So, yes, sir. Let's end the day with divine energy of musical yoga by students from University of Patanjali. Dr. Aarti Pal, Assistant Professor, University of Patanjali, will introduce her yoga team now. Thank you.
Om. My pranams and obeisance to the divine feet of Param Pujya Yogarshi Swami Ji Maharaj and Param Shraddheya Acharya Shri Ji Maharaj, and also my greetings to the delegates of today's function, the divine audience of today's program, and all dear brothers and sisters. I humbly request all of you to please take your seats, because now you are going to witness the energy and the power of yoga. And I take an immense pleasure to invite here the Yogasana sports team of University of Patanjali, which is an institution which aims at holistic development of the students. The students of University of Patanjali are going to present here today an artistic Yogasana performance. Artistic Yogasana performance is a form of Yogasana belong to different categories of asanas, where forward band asanas are included, backward band, stretching asanas, twisting, and many others categories of asanas are there. I would specially like to mention here that artistic form of yogasana or these different forms of yogasana and also the inclusion of yogasana as a sport is the virtue of the initiatives taken by our Pujya Swami Ji Maharaj and Pujya Acharya Shri Ji Maharaj. Under the guidance and the initiative of our masters, the World Yogasana Sports Federation has been formed to spread the yogasana at world level and also to popularize and prioritize yogasana as a sport at national level. NYSF, that is National Yogasana Sports Federation, has been formed. And soon we are going to witness yogasana as a part of Olympic Games as well. So now today, this really indeed needs applause because the knowledge of yogasana of ancient India will get the recognition at international level. So now I am inviting here the girls team of University of Patanjali. They are going to perform an artistic performance on Lord Shiva theme. Lord Shiva is known as Adi Yogi, the master of or the originator of Hatha Yoga. Most of the asanas that you witnessed today have been explored from Hatha Yoga. So now let's welcome the girls team of University of Patanjali to witness a wonderful performance. And now the boys team of University of Patanjali is coming here with a message of spreading peace and harmony by yoga at the world level. Please welcome them with a big round of applause.
विश्व में शांति रहेगी योग से, विश्व में शांति रहेगी योग से, मन की भी ज्योति जगेगी योग से, मन की भी ज्योति जगेगी योग से, विश्व में शांति रहेगी योग से, विश्व में शांति रहेगी योग से। आपका अभ्यास जब बढ़ जाएगा, आपका अभ्यास जब बढ़ जाएगा, ध्यान की शक्ति बढ़ेगी योग से, ध्यान की शक्ति बढ़ेगी योग से, मन की भी ज्योति जगेगी योग से, मन की भी ज्योति जगेगी योग से, विश्व में शांति रहेगी योग से, विश्व में शांति रहेगी योग से। धीरे धीरे रोग भी मिट जाएंगे, धीरे धीरे कष्ट भी मिट जाएंगे, विकल्प रहित संकल्प बढ़े रहित संकल्प बढ़ेगी योग से, मन की भी ज्योति जगेगी योग से, मन की भी ज्योति जगेगी योग से, विश्व में शांति रहेगी योग से, विश्व में शांति रहेगी योग से। जो ग्रहों की चाल को भी दे बदल, जो ग्रहों की चाल को भी दे बदल, ऐसी शक्ति भी जगेगी योग से, ऐसी शक्ति भी बने।
Thank you. Thank you all the dignitaries and eminent guests for your time and making the day one of this conference conclude successfully. Thank you to all the HODs. Thank you to all the students for having their patience and inquisitiveness and taking so much interest throughout the session. We will meet here again tomorrow at 9 a.m. to take the science further to the next level.